see this strip. Okay. Huzzah. Okay, well, anyways, welcome to the Intro to Biology workshop. I think we are actually going to be able to cover a lot. There's sort of this misconception that, like, if you go into a biology course, first thing they do is have you memorize just piles of just garbage. The reality is we have literally all of human knowledge in our fucking pockets. There is no reason for that. Literally the majority of the university degree that I did do was memorize these tables, which you can look up, like memorize these structures, which you'll never need, like memorize all this crap. Useless, not important, doesn't help anybody at all. So what is important is to understand that biology is just really complicated chemistry. There are some really straightforward rules, but biology is basically the closest to like alien technology that we have insofar as we didn't make it. We have to figure out the rules the hard way. Whereas with a computer, fundamentally, there is a book that will explain every single part of it that you could ever want to learn.
Unless you lost the book.
Unless you lost the book. But yeah, so when it comes to biology, there's a few like core aspects of it that are important to understand. You don't need to know them in excruciating detail unless you're doing something that directly involves those. So my experience with biology is that every single time I've wanted to do something, I can just learn it when I need it. There's just too much. Like any one organism has more detail to perfectly describe how it works than you could ever cram into any book or brain ever. It's just overload, but you don't need it. There's also a lot of different areas to biology, so depending on what you want to do, you can kind of specialize very, very easily, and you don't need to know all the rest of it. So there's even just different types of, even between species or types of thing, there's huge variation in how things are handled. But what we're going to be going over is the kind of core shit that happens in everything, because that's really the important stuff. Like once you have this down, then you can just kind of go in and like fine tune what you know. Okay, so big question, what actually makes something alive? Now, when I was in university, this is the sort of basic definition that they gave us, which is it's made of cells. Cells are the fundamental unit of life. No cells, no life, generally. But I'll talk about that in a second. Produces waste. This is sort of this goes really well with eats. Basically, the main thing that makes something alive in the traditional definition is that it can vastly control the inside of itself whilst interacting with the outside of itself. So it, very low entropy on the inside, very high entropy on the outside. Very high complexity on the inside, not so much on the outside. Basic idea. The replication is also a big one. Every cell in every one of your bodies did come from one individual cell at one point. And I'm not talking like sperm. I mean, like, thanks to evolution, literally through billions and billions and billions of years. And we'll talk about this in a second. There's what's called LUCA, which is the last universal common ancestor, which is the bit of goop that everything came from. And we can actually trace it back generally to a thing, but what that thing looks like is kind of iffy. But yeah, so that's the traditional definition. The modern long answer is that it's a little more complicated, primarily because of things like viruses, because viruses don't do any of this. They cannot exist without other cells. And yet, they replicate, they spread, they take in stuff, they do stuff. They're kind of a weird case because they don't have a lot of the internal machinery, they rely on other things. But if not being able to survive on your own is what is a cutoff, then huge portions of biology would be cut off from also being alive. Because there's tons of different parasites that cannot exist without their host. So this
like
is why
symbiotic twin.
Right. But more like epiphytes, like plants that grow on other plants. They do not exist if they don't have a tree to grow on. So if that's the line, it's a weird way. It's a weird line. And then now it's getting even more complicated with the advent of computers of ridiculous complexity. So now you're having viruses that behave, by all counts, the exact same way that the natural variants do, which is weird. Exactly, but it's not alive. But is it? I don't know.
How do you? Yeah.
Topic for philosophy that I frankly don't care about. Because it doesn't matter. Like I tend to stick with the things that work in the traditional way just because it's convenient. Okay, so this is your basic tree of life. Basically, everybody draws these differently because there's like the one that I had to memorize, and I do mean memorize, for my second year bio class. Bastard made me memorize every single one of these and the properties of each. Fuck that guy. But that said, there are basically three main hunks to this that are pretty consistent. So the blue is bacteria. We're all fairly familiar with bacteria. I hope they're everywhere. Everything you've ever interacted with is covered with bacteria, almost definitely. There is a huge variety. Like we have maybe discovered 0.1% of the bacteria that exist. There is just infinite variety, basically. So there's a couple really common ones. So things that you might think of as something else are actually just bacteria. So most algae, bacteria. So specifically cyanobacteria. So like blue-green algae is literally just shitloads of bacteria. But again, there's bacteria everywhere. You are about 3 pounds of bacteria. There are more bacterial cells in you than there are you cells in you. They're just much smaller. So there's bacteria. Then there's this weird hunk over here, which are archaea. Originally, these were also stuck in the bacteria bucket, but they're really weird. They're actually a lot closer to us than they are to bacteria. Maybe not us specifically, but the greater. We're going to cover this in a second. There's basically two types of cell: the itty bitty ones that have no nucleus, and the bigger ones that do have a nucleus. They're closer to the ones that do have a nucleus for a variety of reasons. They're also some of the weirdest. They tend to live in ridiculous environments. The stuff that lives in just like crazy hot or cold or salty conditions tend to be archaea. They're weirdly robust. And then basically the red bit is everything else. Your plants, your fungi, your animals, your algae.
Right.
Some
Did you
algae.
say algae was part of bacteria?
It is. There's different kinds. So blue-green algae specifically is bacteria. There are other algae that are, the cells are bigger, or they arrange in weird ways, or are weirdly multicellular. There's a lot of variations. With biology, the rule is there's very rarely an actual rule. There's always exceptions, which is always kind of fun.
So basically quantum mechanics.
I mean, yeah, it's basically goop powered by quantum mechanics and chemistry.
Oh, that's true.
And a lot of physics and a lot of chemistry, but we'll get to that in a second. Okay, so these are your basic cells. So on the left, the big one is what's called a eukaryote. Basically, it means it's got a nucleus, which is this bit here. If you've ever cracked an egg is just one big ass cell. The yolk, nucleus, vaguely. There's a whole bunch of different bits that go into this. The other type is the prokaryote, which is your bacteria, much smaller, no nucleus. It's basically just a tube with shit floating around. So let's look at eukaryotes first. We're going to mostly be talking about bacteria today, just because they are the easiest to work with. But it's important to know what's going on here. So there's a couple different main pieces. The nucleus is where all the DNA is stored. DNA, instructions for life, everything that tells your cells what to do lives there for the most part. This roughly bit on the outside is called the Endoplasmic Reticulum. Very fancy name. It's basically just folds that a lot of the protein making machinery, which we'll talk about in a moment, lives there. Like post-processing stuff as well. So there's this something we'll talk about in a little bit is that proteins are basically what make everything work. They are the teeny tiny machines. The general way that it's taught is that you have one gene, Gene makes protein, and then that's just kind of it. There's a lot of actual like post modifications that happen in proteins before they're actually ready to work. So all of that kind of extra modification happens there. Think of it like it's your your auto shop, like it's your detailing shop. Like you want spinning rims, it happens in the endoplasmic reticulum. So then you've got the Golgi Apparatus. This is your mail system. If you want to import or export anything, it goes through there.
Let's hope they allow biological material.
Basically, think of it like the alien that's like smoking in the mail sorting machine in Men in Black. It's that. So it does any packaging that stuff is going out of the cell goes through there first. It'll do extra little modifications as well. It's also able to basically check stuff and figure out if it's supposed to leave or go or what's supposed to happen to it. The next bit are the mitochondria. They're these guys. We're going to look at them more in a moment. They are where all of your energy comes from, basically. They're actually really weird. They're actually bacteria that millions and millions and millions, billions of years ago, some cell ate one. But for whatever reason, it didn't kill it.
It's like, I don't know these things.
Yeah, and so there's all sorts of really interesting research on studying how this happened, because this actually happened twice. Once for mitochondria, and then a second time in plants for chloroplasts, which are what make plants green and photosynthetic. We'll talk about those in a second. Mitochondria, you will hear this at every bio lecture you will ever go to. Mitochondria is the powerhouse of the cell.
Oh, God,
it's been said, we're never saying it again. There is a ton of chemistry that goes into those, whereas a very, very small amount of the energy processing happens outside of them, and it basically just like takes those simple precursors, like it goes from like sugar to a very simple precursor that it just pipes into the mitochondria as fast as it physically can, and then the mitochondria is way more efficient at extracting energy from it. So all of that happens there, and the mitochondria basically just like bleeds energy molecules, which is really quite handy. They're also very oxygen dependent, so this is why your body is not so happy if you don't have oxygen, because they require it to work. So moving on, this one's a weird one. It's called the Centrosome. It's basically, we don't really know how it works. It's weird. It is a complex of like hundreds of proteins. It looks weird. It looks like pasta. But coming off of those, there's two of them actually. Coming off of those are microtubules, which we will look at very briefly in a moment. Cells actually have a skeleton to them, made of little fibers of different proteins. There's a couple different types. They do different jobs. One of them are microtubules, which hang off of the centrosome. They're basically in charge of organizing the cell, and when the cells go to divide, which we'll talk about in a second, they play a major, 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 major role. Then there's just a couple other little bits. These little red things are called ribosomes. They're what actually make the proteins. So they read RNA. So first, we're going to talk about this in a second, but DNA is translated into RNA, and then RNA is fed into the ribosomes, and then they turn that into protein. Like they just read out the code and go, okay, I need this thing, and then this thing, and then this thing, and then this thing. It's a very neat little system. And then just the last couple of things, we've got peroxisomes and lysosomes. These are basically just little bubbles that are full of stuff that can break things down. So any like really hardcore digestion happens there. Or if they eat something, like say this is a white blood cell, if you're going to break down some bacteria you just ate, you just shove it into a thing full of basically hydrogen peroxide and just rips the fucker apart. Yeah, kind of metal. I like it. Bacteria are way more simple. A lot of this isn't there. They're literally just tubes with stuff floating around. Way less organized, but also way smaller. So they don't have a nucleus. Their DNA is not organized neatly. It's organized in basically just a tangle right in the middle, but it just kind of floats around.
I thought it was spaghetti.
It's life spaghetti, basically. One last thing. In all cases, this outer bit here, we call the plasma membrane. It's basically made out of oil. Like all cells are basically just oil droplets with water in them. Really quite weird. There's a lot of interesting stuff that can be said about this specifically.
Battery's running.
Okay, give me 2 seconds. I'm gonna grab my charger. This whole time I'm like, I feel like I'm forgetting something. I can't imagine what it is. Oh, wait. Computers require energy too. But yeah, so plasma membrane, lots can be said about that. Frankly, too much to cover. But just know that it is basically the boundary of what keeps the insidey bits inside and the outsidey bits outside. There is a huge amount of proteins that are embedded in that that control what can come in and out, basically, which is a workshop in and of itself. Bacteria are a little weirder. Sometimes they have extra layers on top of the plasma membrane, just as like extra cushioning. They're very small and delicate, so a little bit of extra shell is helpful. Plants also have an extra shell, but instead of it being all the weird shit that bacteria are made out of, it's made out of cellulose. It's also what makes them extremely rigid. That's why plant cells legitimately do look square or like polygonal. It's that cellulose. It makes it extremely rigid. The more of it they have, the stronger they are. They also can have another material, which is lignin, which is what makes things woody. So it's all these different things are just shell, basically. But yeah, so that's the cell, the very, very basics of what life are. So once you've got one cell, you got to make more, because what's the good of one cell when you can have a billion? So there's a whole bunch of different ways that this can happen. Depending on if it's a bacteria or if it's like a eukaryote, there's two main ways. So with bacteria, they tend to just copy their DNA and then split in half. It's called binary fission. There's also budding, where they basically just like grow a little tumor-looking thing and just like slowly fill it with all the stuff that it takes to make that into a cell. And once it's fully packaged, it just lops it off, and then you've got a new cell. With eukaryotes, they're bigger. There's a lot more going on. A lot more needs to be copied. Because think about it, every single cell needs to have all the same stuff, or it doesn't work. So to do that, there's this really intricate dance of stuff moving around. So the main way that this happens when it's just a regular cell, most of the cells in your body, it's called mitosis. So there's a few main phases to this. This is what a cell kind of looks like. These are very particular types of images called immunofluorescent images. Doesn't really matter. Point being, this bit is the nucleus. So you can see that it's very clumpy. During most of a cell's life, DNA is not bound into a particular structure. It's floating around because it needs to be. It needs to be uncoiled so that proteins can grab onto it, write, copy, do all its stuff. If it's all coiled up, it can't function. But when it needs to divide, it kind of has to pack all its crap into a box so that it can move. So the way that it does this is first, all of the different pieces of DNA, depending on the organism, will bunch up into what are called chromosomes. They're just discrete bundles of DNA. And so once all the chromosomes are all packaged up into their nice little shape, they all line up around the middle, called the equator. And then those centrosomes, the thing that the microtubules come off of, basically grow onto them, onto the very middle of each of the chromosomes, because chromosomes come in a pair called chromatids, because they've just made a copy, right? That's the whole point of cell division. So they make a copy. Now you've got your two pieces. The two pieces line up in the middle. Centrosome grabs it right in the middle, and then once everything's ready to go, there's all this really complicated like chemical pathways and checks and balances. Like, are we good? Are we good? Are like all lights green? If yes, go. And as soon as go happens, it literally gets pulled apart into the two halves. And then once they're separated, it grows a new nucleus around it, and then pinches off, and now you've got two cells. The other way that this happens is for mating, basically, which is meiosis. Same basic concept, but you end up with too few chromosomes in each of the individual cells, so that the only way that you can get the proper amount is you take one type and the other type, make them shmoo, and then you've got a functioning thing. So in this case, it's typically an egg and sperm. There's some variation. It can get weird, but that's the basics. So yeah, that's cell division. Really quickly, I want to show you what this actually looks like because everybody shows this drawing, but it actually looks really cool because it does do this. Yeah.
What speed is this on?
That's probably over a couple of hours. It's a fairly slow process. But I mean, it literally lines up and then gets pulled apart. You can see the densest part right at the edge here is actually where that centrosome is. We're going to look at what this 3D animation of what is actually going on here in a minute. It's something fascinating. But yeah, so that's that. Yeah, cell division. It's a very, very complicated thing. They tend to make you memorize all of this. It's just important that you know that it happens. Memorizing each step doesn't matter because you never use it. But if you get into bio and you really start messing with the like delicate little machinery, you can do all kinds of weird shit by tinkering with this. Most cancer drugs, maybe not most, but a lot of cancer drugs work by screwing with this process. And grabbing onto various biochemical things that turn the process off. So one of the most common cancer drugs called paclitaxel, which actually comes from a type of tree, because it's only a toxin until it's medicine. Will basically turn this process off. Or no, wait, that's something I'm thinking of something else. Point being, various drugs will turn this off, so that way your cancer stops growing. So that way you can, burn it, cut it out, whatever, and it won't make more of it until you turn it back on. This is why most exactly. This is why the process of treating cancer is attempting to kill the thing before it kills you. Exactly. Yeah, ideally without killing you, but it's a mixed bag. So yeah, there's lots of very interesting stuff going on here. Mostly important that you know it happens. Day to day, you don't really interact with this much. But there are really great experiments that let you visualize this and see it actually happening, which is great. When I say see it happening, I mean you can see the various stages happening. It's a fucking slow process. You can't watch it in real time like we just did. Okay, so like I said, life is chemistry. A lot of it. Like an aggressive amount. So over here is a vague metabolic chart of approximately all of the things that would happen in a cell. You see this little circle right here? That's that. That's one tiny piece. And every compound in there gets used in other pathways and is used in other products, and just everything is used for everything else. This particular one here is called the citric acid cycle. It's the main way that you take in sugar and make energy. And you can also feed this cycle through fats, proteins. It's basically the main engine that keeps everything running. It also happens mostly in the mitochondria. So this is what they're doing. It's an extremely efficient system and sucks every last ounce of energy out of a molecule of sugar that you can get. Also a thing they have you memorize that to date I've never actually had to use. But it is important that you know that it's there, and it's fascinating to watch it, because there's a lot more layers to this than what's shown in that graphic. Each step requires an enzyme, which we'll talk about in a moment. Everything has bits that go in and come out and interact, and everything needs to be perfect for it to work. But it is just a lot of chemistry. As to this fuckery, this is everything else. Every part of your cell is doing some part of this at some point. It's just really complicated. It's just a lot of moving parts interacting with each other. But the nice thing with this is the sheer vastness of the different chemicals that are in there is really interesting, because it lets you, anything you could ever want, there's a good chance that some type of cell makes it. Or makes a precursor to it, or you can like kick it the right way and it'll make the thing that you want.
I want an iPad.
Okay, maybe not that. It's a little harder, but like, you can, everything makes a little bit of like formaldehyde and ethanol and all these, but also like much more complicated compounds. Things like lactic acid, which now we've got PLA plastic, which is just what happens when you take lactic acid and put it in a chain. So if you've got enough of it, you can process it down. So there's all this tons and tons and tons and tons of useful chemistry there, which is very interesting and also very frustrating. Okay, how does any of this shit work? 3 basic steps. You start with DNA. DNA is basically the code of life. There is a lot of variation between how DNA is handled between different types of organisms. But there is some very basics that is pretty much conserved across everything. This is basically it. Anything beyond this basic picture, there's huge variability. But everything basically does this, where you have a piece of DNA, and at some point in that piece of DNA, there's what's called a gene, which is just a stretch that says, this is a thing that you should make. How that's handled varies pretty wildly. Once something happens that tells the cell that it needs whatever that gene codes for, a protein, which is in this case is RNA polymerase, amongst other things, grabs onto that piece of DNA. And it literally reads it, and as it reads it, it copies it into RNA. So let's just quickly go over what DNA is, what RNA is. So DNA stands for deoxyribonucleic acid, which is basically this gray bit is made out of a particular type of sugar and phosphorus. And so that's what's called the backbone. And then the individual letters are these guys here. So there's 4 of them. You'll traditionally see it shortened to AGCT, but it's adenine, guanine, cytosine, thymine. They'll stick together in very particular ways. So A always sticks to T, G always sticks to C. So when the thing is being copied, it writes out whatever the opposite thing is that it's reading. So if it's reading an A, it sticks a T on it, etc., etc., etc. RNA is a little different. It's basically the same. The only difference is thiamine. Instead of thiamine, it's subbed out for a different, very similar compound called uracil. Doesn't really make a huge difference. It's the same thing. RNA is a little weirder because DNA sticks to is double-stranded, right? So all the little chemical bonds that are dangling out are accounted for because it's got a partner to stick to. RNA not so much. RNA is actually a lot more chemically active than DNA is, so you can make all kinds of weird shit out of RNA. My professor in university studied how to make enzymes out of nothing but RNA because they have that level of chemical chooch that they can actually grab onto molecules and do stuff with them. But for all intents and purposes, for the sake of the discussion today, it's not really important. There's a lot more that RNA does. There's all kinds of different types of RNA. They'll put a little label in front of it. So, if it's mRNA, it's messenger RNA, which is this stuff, which we'll talk about in a sec. There's also tRNA, which we're not going to talk about today, but is very, very important for this process. There's also small interfering RNA. There's all these different variations. Because it's single-stranded, it can basically stick to other pieces that are floating around. So one way of turning stuff off is just plug up the process. So if you can just make a piece that matches, it'll stick, plug up the process, and it can't work. Because for RNA to be copied into protein, which is the slow-read bit, it requires that nothing else is attached to it. If there's anything in the way, it stops the process. So a lot of chemical, cellular control is controlled that way. Where just stick a thing to the RNA that's supposed to be copied and it stops working. So then from mRNA, which is messenger RNA, then we can actually make proteins. So proteins are just long strands of amino acids, which I'll talk about in a second. They're polymers, basically. And based on which amino acids you put one after the other, vastly change the properties of the final thing. But this is basically it. This is what's called the core dogma of biology, basically. So you go DNA, RNA, protein. It always goes that way. Exactly how this is handled and everything is a little different, but this is always the way everything does this. Bacteria, eukarya, everything does exactly this, because this is how you make everything that works. For the most part, proteins are the tiny little machines that make everything function. life is kind of weird. It's basically a collection of dead things, i.e. proteins, that if you've got enough of them, it makes it alive somehow. I'm not really quite sure how, but it's some level of complexity is what actually like tips the balance, and you end up with life somehow. Okay, so let's look a little more closely at proteins. So as I said, they're made of amino acids, which are just different chemicals. These are all of the amino acids that are traditionally used in most kinds of life. They're not the only amino acids because it's possible to make synthetic ones that have different bits, but they all have the same basic pieces. So they've got a nitrogen group on one end, and then they've got a carboxylic acid on the other. And then a little dangly bit that changes the properties. And so depending on what the dangly bit is, changes what happens when you use these. So for example, this one, I realize this is hard to read or impossible. But this one's called cysteine. That one has a sulfur group hanging off of it. If you've got two of those in a protein, the sulfurs will actually stick together. So it's one way of bending a thing and holding it in place. So keratin, the stuff in your hair, the reason that your hair takes a particular shape is because of a copious amount of those that hold it all together. This is also why burning hair smells so goddamn bad. It's all that sulfur coming out. And also all of the nitrogen and nitrogen compounds coming out, but largely the sulfur. These are kind of broken up into polar, non-polar, hydrophobic, hydrophilic. Basically, it just means how they like to interact with the stuff around them. Do they like to be in things that are more oily, or do they like to be in water? And the way that proteins know how to fold, which is this next bit we're about to talk about, is largely because of that. Certain functional groups, the dangly bits, really hate being in water. So they'll all stick together and like huddle around in the middle, so they're as far away from the water as they can physically get, which is really quite interesting, actually. But all these different things can do different things, and there's no really good, well understood pattern of, what goes where and why it works. Figuring out how to make novel proteins is damn near impossible. We can do some very basic stuff, but fully designing a protein from the ground up, nobody's done well. It's extremely hard, which is why we tend to start with or find natural proteins and then just fuck with it until it does the thing we want. So moving forward, there's basically 4 layers to a protein, right? So there's the primary structure, which is the actual sequence of amino acids. Then there's the secondary structure, which is the very basic ways that it folds, and so it tends to do this in one of 2 or 3 ways. We've got beta sheets and alpha helices, so a spring or a sheet. The sheets tend to be much stronger. There's a lot more bonds holding them together and holding it to each other. It's more like a crystal than anything. Whereas the alpha helices are springy. So the project that I'm working on is spider silk. So the reason that spider silk is so strong is because it's these huge areas of that highly crystalline beta sheet held together by the stringy bits. So you get this really nice mix of both extremely strong and stretchy, so that's why it's so strong. But these can bend and twist and make into all kinds of weird shapes. So then the tertiary structure is what happens when each individual protein is completely folded, right? So it's this mixture of helices and sheets and other random assorted bits all coming together into a shape. But not all proteins, in fact, not most proteins, don't exist on their own. They tend to stick to either exact copies of themselves or to other proteins. So that's what's called the quaternary structure. So it's basically how individual proteins fit together. So this is one example where you've got 3 identical proteins that will stick to each other in one big chunk. But lots of things are not like this. They're made of all kinds of different ones that all fit together like Lego. This is basically just squishy Lego. Squishy catalytic Lego. So this is kind of a very basic idea of just the ridiculous diversity that you get with proteins. You can have big ball stuff like this. So this is a virus, which each one of the different colors is there to represent a different protein. There's half a dozen in there, and they all fit together perfectly that through repetition, they end up with this perfect sphere. But you also have stuff like this, which is an antibody, or just random other bits. This is very likely a protease. So once you've got protein, you've got to do something with it. Otherwise, it'll just clog up everything. So stuff that the cell doesn't need anymore will get fed into something like a lysosome. Or there's enzymes called proteases, which will grab onto proteins that are floating around and just shred them back into their individual pieces. Yeah, so it's like a cycle. It's like a recycling plant, basically. Once a thing is done or isn't needed anymore, it gets shredded, destroyed, and then other things take over. And there's this constant flow and balance of things being created and destroyed to keep you functioning, basically. Yeah, proteins are extremely various. Yeah.
So what was the tertiary structure?
Tertiary is the structure of each individual protein.
So yeah. A single protein can have fold in some protein sheets and some protein.
Exactly. Yes.
Okay. Cool.
When you were doing the genetic modification to attach the silk-producing proteins to the bacteria, it's between the tertiary and the.
That one's
this is where you were kind of placing it so that the final protein would.
Kind of. That's sort of a longer discussion. There's more to that.
Okay.
I can take a break if someone wants to grab it. Thank you. While he grabs the door. So we're going to do a whole workshop on genetic engineering itself, so that way we can really deep dive into that, because I don't know when we're going to do it yet. We're planning on it. We're getting through the month, maybe next month. So that really requires looking more deeply at the way that DNA is handled, because there is some protein engineering that we're doing, but it's very basic. And this is kind of the general theme when it comes to protein engineering. We can't make individual proteins, but we can Frankenstein them together. And so you can take like the bit that actually does the work from one protein and the bit that does the work from another protein and just smooth them together into one big protein, and it'll do both. So for the spider silk thing, we've basically got this really long stretch that's just the silk bit. And then we're taking the other functionally bits and just shoving it at the end. And so when the DNA is read and made into mRNA, it's all made into one long piece of mRNA. And so when that's read and turned into protein, it's just made into one long amino acid sequence. And then it all folds, and so the silky bit folds into its usual silky shape, and the bit on the end folds into its usual shape, but they're stuck together physically, because they're all one long protein. So that's how you can get like added functionality, basically.
Okay. But the ideas of single protein always folds the same way, or
for the most part, yeah.
The same way?
Usually. That kind of gets into things like Parkinson's and Alzheimer's,
right,
where if it folds wrong, that's a big problem. Hey, how's it going? Yeah, it's all good. Grab a seat. Okay. But yeah, so proteins, this is sort of the weird thing, right? Like a protein can have thousands of amino acids in it, and yet it seems to consistently always fold the same way, which is really weird. Part of the way that it's able to do this is because of all that post-processing and packaging, but a lot of it is just because of the things like the hydrophobic bits. The hydrophobic bits always are hydrophobic. They always want to be away from water. So as basically as a protein is made, it immediately starts folding. It doesn't like wait until the whole thing and then like folds, because that's when we simulate it. That's how we tend to simulate it, where we just take the whole, like we know what the string is, and so we like plop the string into a simulation and just go fold. And then we're surprised that there's like 10 to the 300 or whatever different ways of it potentially folding. This is why it's actually really hard for us to know the shape of a protein, and why there's whole fields of science designated to studying this, because it's very consistent in nature, but it's also very hard to simulate just because of the sheer like difference in all of that. However, for whatever weird reason, human brains really good at figuring this out. Computers, absolute garbage. So there's this game that you can play called Fold It, where basically you're handed a protein where we kind of know the way that it folds. Like sometimes you're given, different data that scientists have collected about that protein. So for example, maybe like an electron cloud. So we've shot electrons at the thing and mapped out where they bounce off. And so we've got this like vague blobby shape of like the protein approximately fits into this blob. And then here's a protein sequence, fold it until it fits into the blob. And so through multiple players playing this game, we actually figured this out and get damn near exactly what the protein is, just through the weird like collective knowledge of people fucking with the thing until it works. Yeah. And so like the best players get credited in the papers that are written about those proteins because you've actually done the work of figuring out the shape of the thing. So like there have actually been papers on HIV and just all this different stuff that folded players have figured out, which is just mind-boggling. But yeah, so a lot of it is, we know these basic rules, your primary, secondary, that kind of thing. But for the most part, it's kind of iffy. So actually figuring out the shape of a protein is an enormous amount of work. I mean, hell, even just figuring out the sequence of a protein can be an enormous amount of work because sometimes you know the protein, but pulling it apart into the individual letters is a huge pain in the ass, and the machine is very expensive. So like even working backwards is hard. So there's a lot of different tips and tricks. And so really, I mean, this is one of the reasons why I maintain that science should be taught as vocational training, like teach you how to use the machines. Memorizing all of this, not helpful. Knowing how that you can pick this apart and figure it out, that's actually what's helpful. But yeah, so this is kind of proteins at a glance. Proteins tend to be divided into a couple main categories. So there's structural proteins, which are things like keratin, like hair, nails, or elastin, which are like what holds your cells together. All that kind of stuff. Then there's transporters, which actually sit in that plasma membrane that we mentioned way the fuck back. These are things that will sort things that are outside and bring them in, or things that are inside and bring them out. Most of the time, and then those are broken into like passive and active. So some of them have a toll. So you pay the toll, a thing will move. So this is a really convenient way of moving things where they don't want to go. Because one of the like core concepts here is like diffusion and osmosis and gradients. Everything works on a gradient. There's always a higher concentration of something somewhere else, and it wants to even out. So if you've got a lot of something outside and very little something inside, it wants to come in. But if you pay that toll, you can keep pumping more of it out and keep the level really, really low. So that's just one way that happens. And then the other one is enzymes. There's also signaling stuff, but that's sort of its own weird bucket. But enzymes are the actual, like, they do all the chemical work. So anything that breaks stuff down or puts stuff together is an enzyme, basically. So really common one, lactase. That one will take lactose, which is 2 sugar molecules, and rip them apart. And it's really interesting watching this happen because it's extremely physical. Like all enzymes really are is this weird gloopy thing with a hole in the middle that fits a very particular compound. So for lactase, there's this spot called the active site. Lactose fits in, and then you just see the whole protein like flex and like get really close and literally go and just literally pull it apart. This is one of the reasons why they're able to do extremely hardcore chemistry at basically room temperature. They're what's called a catalyst. So they participate in the reaction, but they're not consumed in the reaction, basically. And they can work very, very, very quickly. The peroxisome that I mentioned very briefly can process, it's like an absurd amount of peroxide per second. I think it was like 100,000 molecules of peroxide per second. It chuches real fast. We're going to look at a DNA copying enzyme in a second. It turns at about the same speed as a jet engine, but it's very tiny. And it doesn't wear down, which is weird because it can't. It's just atoms. It can't wear down an atom.
Maybe we can make a mini plane, microscopic plane.
People have tried. But yeah, so this is proteins. I know there's an enormous amount here. There's way more than I could possibly cover in any reasonable amount of time. But know that these are the things that do most of the work in a cell. Okay, this one's kind of fun. So now that you kind of have the basics, we're going to watch this whole video because it's just so goddamn fascinating. Now that you have the basic idea of like what a protein is, let's just watch what it looks like. Right, come on, play nice, please. Come on, there we go.
These are tiny molecular machines, and they are doing this inside your body right now. To understand why, we have to zoom out. Every day in an adult human body, 50 to 70 billion of your cells die. Either they're stressed or damaged or just old. But this is normal. In fact, it's called programmed cell death. But to make up for all these lost cells, right now, billions of your cells are dividing, essentially creating new cells. And that process of cell division, also called mitosis, well, it requires an army of tiny molecular machines. So let's take a closer look. DNA is a good place to start, the double helix molecule we always talk about. This is a scientifically accurate depiction of DNA created by Drew Barry at the Walter and Eliza Hall Institute of Medical Research. If you unwind the two strands, you can see that each has a sugar phosphate backbone connected to the sequence of nucleic acid base pairs, known by the letters A, T, G, and C. Now, the strands run in opposite directions, which is important when you go to copy DNA. Copying DNA is one of the first steps in cell division. Here, the two strands of DNA are being unwound and separated by the tiny blue molecular machine called helicase. Helicase literally spins as fast as a jet engine. The strand of DNA on the right has its complementary strand assembled continuously, but the other strand is more complicated because it runs in the opposite direction. So it must be looped out with its complementary strand assembled in reverse, section by section. At the end of this process, you have two identical DNA molecules, each one a few centimeters long, but just a couple nanometers wide. So to prevent the DNA from becoming a tangled mess, it is wrapped around proteins called histones, forming a nucleosome. These nucleosomes are bundled together into a fiber known as chromatin, which is further looped and coiled to form a chromosome, one of the largest molecular structures in your body.
And there's lots of really great animations that do this sort of thing
You can actually
and
see
accurately show
chromosomes
what
under
this
a
looks
microscope
like.
in dividing
Fascinating
cells.
to watch.
Only then do they take on their characteristic shape. Otherwise, the DNA is more strewn inside the nucleus. The process of dividing a cell takes around an hour in mammals, so this footage is from a time lapse. You can see how the chromosomes line up on the equator of the cell. Now, when everything is right, they are pulled apart into the two new daughter cells, each one containing an identical copy of DNA. Now, simple as this looks, the process is incredibly complicated and requires even more fascinating molecular machines to accomplish it. So let's look at a single chromosome. One chromosome consists of two sausage-shaped chromatids, containing the identical copies of DNA made earlier. Each chromatid is attached to microtubule fibers, which guide and help align them in the correct position. The microtubules are connected to the chromatid at the kinetochore, here colored red. The kinetochore consists of hundreds of different proteins working together to achieve multiple objectives. In fact, it's one of the most sophisticated molecular mechanisms inside your body. The kinetochore is central to the successful separation of the chromatids. It creates a dynamic connection between the chromosome and the microtubules. For a reason no one's yet been able to figure out, the microtubules are constantly being built at one end and deconstructed at the other. While the chromosome is still getting ready, the kinetochore sends out a chemical stop signal to the rest of the cell, shown here by the red molecules, basically saying this chromosome is not yet ready to divide. The kinetochore also mechanically senses tension. When the tension is just right and the position and attachment are correct, all the proteins get ready, shown here by turning green. At this point, the stop signal broadcasting system is not switched off. Instead, it is literally carried away from the kinetochore down the microtubules by a dyning motor. That's the walking guy. This is really what it looks like. It has long legs so it can avoid obstacles and step over the kinesins, molecular motors that walk in the opposite direction. Personally, I'm astounded by these tiny molecular machines, how they're able to routinely and faithfully.
So I can actually do the thing. Come on. Yeah, I'll go back. Right. Oh, there we go. But yes, that's what that looks like, which is crazy when you think about it, right? Like it's literally tiny machines that like can walk. There's a lot of other really great animations that show that in different detail, show other things in different detail. There's one really great. I think it's called the inner life of a cell. There's this whole series of them. One dives really deeply into mitochondria. So that's the citric acid cycle, the thing I mentioned very briefly, the thing that takes in sugar and poops out energy. The very last step of that, the thing that makes the energy molecules, is literally a tiny water wheel. But instead of being run on water, it's run on proton flow. It literally turns in one particular direction, and stuff flows through it to power it. It is so strange looking, but it works really, really well. So this is one of the reasons why I'm such a bio nerd, because it's like you're full of like everyone's like nanotech is gonna save the world. I'm like they're called fucking bacteria. But yeah, so that's that. I figured you'd probably enjoy that. Hopefully, I don't get copyright claim for that, because so that guy's channel is Veritasium. He does phenomenal stuff. If you haven't seen him already, would highly suggest watching his stuff. Okay, so big question: why should you care? Why is any of this important? Like, why bother learning any of this? I mean, other than because it's fucking awesome. So here's just a couple of examples. So this top one is from a company called MooFree. So most of what makes milk is a protein called casein. So fundamentally, there's no reason that you need a cow to make casein. So what these guys are doing is they've taken the DNA for casein and some of the other milk proteins and fats and that kind of stuff, and they're producing it in yeast. So they're basically growing cow milk without the cow, which is just so neat. There's a bunch of other examples of this. So this lower one is BioBrick. So basically, you take sand and you take pee and a particular type of bacteria and some calcium, basically, and it will rip the urea, the pee apart, which the urea specifically that's in it, make carbonate ions. It'll pick up all the calcium, take calcium, carbonate, shmoo, and now you've got basically chalk or concrete, depending on how you look at it. And it knits all of that together around the sand, and you end up with a goddamn brick, which I just think is so neat. But there's all kinds of stuff like this. Another one, so these guys over here are horseshoe crabs. That blue stuff is their blood. Kind of very valuable, like unbelievably valuable. The reason being is there's a compound in their blood that is the most sensitive bacterial test that we know. So if there's even one bacterial cell, it clumps and coagulates. So normally we have to collect and bleed these crabs to get that molecule, which is horrifying. And also it's destroying their population, because unsurprisingly, things do not like when you take them out of the water and bleed them for a while. Who'd have thunk? So there's a company now that's using genetic engineering to produce that compound without having to bleed crabs. So this one up here is one of my personal favorites, just because I'm just a huge nerd for this one particular professor who does phenomenal work. Her name is Neri Oxman. Does some of the finest just stuff. It's awesome. That particular thing is like a dome made from silk that silkworms spun without having to boil any cocoons. So they figured out that if you put a silkworm on a flat plate instead of the usual like meshy goodness, it'll spin a flat cocoon. So once they realized this, they started doing computational design to spin artificial silk on so it's basically like a box or like a frame with little pegs on it, and then they would take a piece of like string basically and just wind it in a particular pattern. And if they got the pattern just right, instead of spinning cocoons, they would spin sheets. So they could just cover this thing in silkworms and just spin a sheet. Then they figured out, okay, well, silkworms like a particular temperature, so they'll migrate to that temperature. So they mapped how the sun was going to affect this thing, poked holes in it so that the sun would distribute the heat evenly, so they could distribute the silkworms. So then they could just cover this thing in like, thousands of silkworms, and they just spin an entire dome without having to boil any cocoons. And of course, they're still healthy, so they lay a ton of eggs after all of this, enough to make something like 20 more domes, because it's alive, it's bio. You feed it, and it makes more of itself. Then of course you've got your glowing plants, which everybody likes glowing plants. And the one over in the corner is actually the mead that we made for Halloween, which is actually, we still have some on tap. So at the end of all of this, if you light it up, there should still be some left. But yeah, so this is why you should care. Biology lets you do damn near anything. All you have to do is be willing to put in the time and the effort, and like literally the world is your oyster. If you just want to make like really colorful flowers or polka dot flowers, do it. Totally possible. It's just going to take a lot of work.
I mean, you can just do that with a marker.
I mean, you could do it with a marker, but growing blue roses is just way cooler. Or anything else you could ever want to make. If there's, make yeast milk, or whatever else. If it's a protein, you can just make it in something else. It might take a little bit of work, because Emerson Bio is easy, but there's infinite possibility. Anything you could ever think of that you want to do, you can do. Case in point, have you ever heard of slime mold?
Yeah.
Yeah, really weird thing. It's actually technically one big cell. And it kind of thinks. And there's an entire group dedicated to figuring out how to do computing with goop. to that end, they've come up with a goddamn coding language for goop. But they very simple, put them between two electrodes and it's a memristor, a thing that people have been trying to make out of silicon for ages. You can make out of goddamn slime mold, which if you've got memristors, you can make anything. So yeah, you can.
What is a memristor?
It's somewhere. It's basically a resistor with memory. So based on the voltage you put through it, it'll respond in a particular way. So you can set it like a bit, basically. But that means you can make AND gates and all the various logic gates. You can do all this kind of stuff, and it's just goddamn slime mold. You just feed it, which is, I just think it's just bananas. But this is why you should care. It's not that hard. You just got to tinker with it a little bit. But the world is your oyster at this point. So yeah, this is why you should care. And then the big question: how do you actually get started? Thing is, you're surrounded by things that are alive, not including yourself, evidently. But there's living stuff, and there's ways to get used to working with microorganisms everywhere. You've probably eaten a dozen over the last couple of weeks. Yogurt is made with bacteria. Pickles. Alcohol. Plants. It takes no effort to grow a plant. Also takes no effort to kill a plant, but it's a different problem. But all of these things, get used to working with the live organisms. If you can make wine, you're 90% of the way to being a hardcore biologist. The only difference between making wine is and doing what I do is the reagents are more expensive, and chances of it not working are much higher. But it's all the same shit. You make something for it to eat. You keep it in a place that keeps it happy. So you learn what conditions keep your organism happy, what your organism needs to eat, and at what temperature your organism likes. If you can do that, you're 90% of the way there. The rest is just some lab skills, which are just moving small volumes of liquid around. That's all of biology. If anyone tells you otherwise, they're lying. It's all just moving small volumes of liquid around. And occasionally chasing down a cow. But it's mostly just moving small volumes of liquid around. That's all of your lab skills. But yeah, that's really how you get started. So we're gonna do some hands-on stuff, get you a little bit used to working with microorganisms specifically, just because they're the easiest to work with. Eukaryotic cells are very finicky. They really don't like not being in the animal they came from. So we have developed ways of tricking them into doing this, but it's mostly expensive. So the reason that you start with microbes is because they're already the complete thing in and of themselves. They are very happy to just eat a little bit of sugar you give them and grow, and you can kind of trick them into doing some things for you for a little while. So when we get around to doing the genetic engineering workshop, we'll actually learn how you do that, like how you start from I have yeast, how do I make it, how do I like trick it into producing a thing that I want. Very, very simple ways of doing this. We'll talk about how you take DNA apart and put copy and paste and put it where it needs to go. But that's kind of a lecture in and of itself.
Wait, so Control C, Control V don't work?
No, it's moving small volumes of liquid around. It's all moving small volumes of liquid around. This is why whenever like a film crew is like, hey, can we come film in your lab? I'm like, you're just gonna watch me move small volumes of liquid around. It doesn't look like anything. Like when Netflix and Vice were filming at the lab, they're like, what can we film? And we're like, look, you can watch us take some red goop out of that and put it into this, and like we can look at it under a microscope. That's it. And like opening the incubator and closing the incubator. That's it. That's all there is to see, because it doesn't look like much, because the things are tiny. That's the nice part about biology. It's fundamentally the tiniest things you could ever get your hands on that can grow into things the size of a goddamn country. Like, it only takes, one algae cell to grow into an algae bloom that's hundreds of kilometers wide. Over significant time and with the right conditions, obviously, but it is possible, right? Like a seed is only this big, but can grow into a whole tree. That's the nice part about biology. It's alive. It can constantly keep doing things until you get to some endpoint, or someone cuts it down. Yeah, so that's that. Okay, so before we get into activities, questions? Anything? No? I realize this was an enormous amount of information that I've just.
I have an obscure one.
Okay.
Synthetic proteins,
Mhm.
they weren't coded and then created. They were created without genes.
No, they're like the coding is the gene.
So we actually, to create like a synthetic protein, like the ones that are rare to create and hard to create, we start by coding them in DNA.
So if you know the sequence of the protein that you want to make, then yes, you basically write it out in DNA. That was one thing I didn't talk about, just because it gets into the really fiddly bits of DNA. DNA is broken into 3-letter, what are called codons. So each set of 3 letters codes for a different amino acid. So if you want to make a protein and you know what amino acids you want, you just write out the codons in order, and then that'll get copied into mRNA, and then that'll get made into protein.
Okay, I've got 3 questions. One, is the stream going to be still online afterwards?
Yeah.
All right. 2,
I know until I get a copyright strike for showing that video.
2, is the PowerPoint going to be shared?
Yeah, I'll link to it.
All right. And 3, we're also going to record the. Okay.
Yep.
You just mentioned it, and I thought it was curious, and whatever. You said that we create false amino acids or whatever, some that are not part of the existing ones. What happens when you feed those in? Do they just kind of take the place of existing ones, or do they
No,
kind of start doing their own?
there's some weird shit that goes along with that. There is actually a way to chemically synthesize proteins. It's not very efficient, so that's why we don't do it very regularly. But also, we've basically taken the enzymes that know how to take RNA and turn it into protein, and we have mutated them to be able to take in new codons that code for those new amino acids. So there is a way to do it. It's just very fiddly.
That's it.
But it is doable. So yeah, it's just a matter of you just need to use a different enzyme, basically. This is generally the answer, like for most of these things, you just use a different enzyme.
I want to know more about signaling, which I'm sure that's a huge topic. But like maybe just basically is all signaling done by making other proteins that manipulate the DNA or RNA?
It varies very, very like immensely.
Okay.
So there's different types of signaling. There's signals that come from the outside of the cell, and then trigger stuff on the inside of the cell. There's internal signaling, but almost all of it works on what's called like a signaling cascade. So for like a really simple one where there's some, let's say a hormone that's floating around in your blood, it will grab onto a protein that's anchored in the plasma membrane. So it grabs onto the outside, which triggers some change on the inside end of the protein, right? So it kind of gives it a tug and the thing moves a little bit. And so then on the inside bit, something else will stick to it, usually another protein. And so then once that happens, then the protein that's stuck to it maybe becomes active. A lot of what will control whether or not a protein is functioning is called phosphorylation or dephosphorylation. So basically, it shoves a phosphorus molecule onto a particular part of the protein, which kind of yanks everything into a different shape and makes it either active or inactive. So if you've got your thing that's dangling on the inside, let's say a thing sticks to it and now becomes phosphorylated. It can now float away and then interact with another protein. And a lot of the times, this one can now hit multiple proteins all at once. So then each one of those can now go and hit another protein. So it's like an exponential growth of stuff that happens from just one molecule poking a thing. And then, down the line, all the way at the DNA, there's different proteins that hang out in the nucleus. One of those in that pathway will come and kick it the right way, which will, trigger a thing. Other times it's, so with lactose, for example, the way lactose works is there's a protein that sticks to the bit of the lactose gene right at the start. It's called the lac repressor. So it'll just stick there unless lactose is present. As soon as lactose is present, it grabs onto the lactose repressor, which causes it to let go and float away. And as soon as that happens, all of a sudden there's no more block there. So the RNA copying enzyme will grab onto the DNA and read it and make RNA, and then it'll do the thing. There's a lot of that kind of stuff where there's repressors or inhibitors, or it'll go the other way where it's a thing that's floating around, and when something sticks to it, it'll then grab onto DNA and start the process.
Right, that's like, these are all proteins though.
Yeah, it's all proteins. And they'll interact with individual compounds, but for the most part, it's all proteins that are doing the work. But like hormones could be a compound, like just a regular normal chemical, that like kick a protein the right way, and then the protein's going to do all the work. Yeah, so that's signaling. There's a lot to it. Like most of a medical degree is just learning all of the weird signaling pathways, because most disease is some pathway got kicked the wrong way, or isn't working, or is overworking, and so all the various drugs are just like turning things up or turning things down or turning things off or turning things on just to rebalance everything. Yeah. Anything else? Cool. Okay, so now the next thing we're going to do is we're going to actually do a little bit of microbiology. So those are what are called Petri dishes or plates. Probably seen them before. Inside them is two things. Something for your bacteria to eat and a gelling agent. Now, you can't use gelatin. The reason being gelatin is actually just protein that you've cooked until it turns into goop. So most things can eat that. So what we use is actually what's called a polysaccharide. So it's basically like a long chain of sugar molecules. It's a very particular kind of sugar. Most things can't eat it. It comes from a type of kelp, I believe. And so nothing is able to eat it, but it's a really great gel. And gels are great because they're full of water. So it provides both the water, because everything needs water. And now you add your food, and now you've got something for it to grow. So I've made some plates in advance, so that way we can play around with those a little bit. We're also going to make some liquid media. So the liquid media is same basic concept, but without the gelling agent. So generally when you're working with microbes, you do one or the other. You're either working with plates or you're working with what's called broth. They call it broth because it's basically chicken soup. In a pinch, you can totally use chicken soup. But yeah, so we're going to be making some broth because I've already made plates.
Can we eat the extra?
You can. It's just it smells like burnt mac and cheese. It's not a great thing. I can't think of anyone who's actually eaten LB and gotten, yeah, delicious. Although I do know someone who drank DMEM, which is the stuff that you use to keep animal cells alive. It's like, well, it's perfectly balanced. It's exactly what your cells need to be alive. True, but also doesn't taste very good. But so there's plates come in a lot of different forms. Depending on what food you use will also affect what grows, basically. So the most common is what's called LB, or Lysogenic Broth, which is basically Marmite or Vegemite, depending on your particular flavor. It's yeast that they cooked and popped open. And so all of the stuff that lets yeast live is now floating around, so other things can eat it. So yeah, we're gonna be using LB plates. They're the most common by far. But you can have other stuff. You can put lactose in there if you want a bacteria that eats lactose, or you can put other stuff if you want to figure out what bacteria you have. So before we had really cheap sequencing, the way that you did it was you run all these different plates that had different things on them, like dyes or particular types of food, and only that particular type of food. So you're able to like sort out what kind of bacteria it is based on what it's able to eat and what grows. Yeah. The dyes are great because some things will produce an acid as they eat, some don't. So if the dye changes color, you're like, okay, well it makes an acid. And so it's basically just like this long process of figuring this out. We're not gonna bother with any of that. We're just gonna do regular, normal, like media. The liquid media we're making is actually gonna be carbon free, and this is for a very particular reason, because the carbon source is going to be styrofoam. So yeah, anyone who's watched my channel has seen the superworm thing,
Yeah.
where basically you take this weird type of beetle larva, which for whatever reason can eat styrofoam. But the thing that lets them do that is the bacteria in their stomach. So I've got mealworms that have been growing and eating nothing but styrofoam for about a month now. So what we're going to do is we're going to prepare the liquid media, and then once it's all sterilized, I'm going to just add the styrofoam after. And then I'm going to have to, once y'all are long gone, I'm going to have to dissect out the gastrointestinal tract of those worms and just drop it in the stuff and just wait for the styrofoam bacteria to grow, basically. But the reason, yeah, I know, it's biology. Everything is a little bit gross.
No, I meant it's gonna be tedious.
Oh, absolutely. I hate doing microscopic testing.
Yeah,
It's the
exactly.
worst. Yeah, try doing that on a maggot. Not fun. Yeah, so that's the first thing we're gonna do. The next thing we're gonna do. Oh, okay, just one last quick bit about plates. So these are some of the like selection plates. So this one is called McConkey's agar. So it'll change color based on if a thing is eating what's in the gel, basically, and it'll also like if something grows, that's also part of the selection. So it's like 2 for 1. This one up top is called Excal plates. So it turns blue because there's a thing that mimics lactose, but instead of it being 2 sugars, it's a sugar and a dye molecule. So if there's something that can eat lactose, it'll break it apart and the dye comes off and then turns blue. So that way you can actually see the little colonies that are blue are the ones that can actually eat lactose. Very simple. And then this one's called the motility test. So some bacteria, not all, actually have stuff that let them swim around a little bit. Some have like a little tail, some have little hairs, some are just kind of weird, and it's somewhere in between. But so this, basically the way that it works is you take some bacteria on a stick and just and stab it into the gel. There's a color changing thing in there, so once it grows, you can see it. But if it stays as just a stab, it's not modal. It can't move. But if the whole thing turns red, it has swum the fuck away. So yeah, that's plates. So yeah, then the next thing we're gonna do is actually streaking, which is how you get this like classic like Petri dish look, basically. The whole point of streaking is to get individual colonies, which are these guys. So the way this works is what you're essentially doing is diluting down whatever bacteria you put on there. So by the time you're at the end, there's actually individual bacteria in each spot. So each one of those colonies started from a single bacteria, and then that's replicated millions and millions and millions and millions and millions of times to make individual colonies. So this is an extremely important skill, especially with genetic engineering, because you always want to start, if possible, from a single colony, so that way whatever you're working with is exactly one genetic makeup. There's no variability because it all came from the same cell. Whereas if you were to take it from the top bit, it's just a random mix of stuff. So it's not as good. It still probably work, but it's better to take from individual colonies. So this is one of the first things you learn how to do when you're in like a micro class. Super handy. It's also really good, like if you've got a mixture of different stuff, because you're diluting the individual cells, it's a way of separating them out. So that way, if you've got 10 different things in your solution, you can just streak it out and then go, I want the red one, because it'll just be like one little red color. So yeah, we're going to do some streaking. And then the last one, which hopefully we have time for, is called gram staining. This is basically a way of breaking bacteria into the main two groups of bacteria. Because basically the way it's divided is what they use as a shell. So gram positive use one type of shell, gram negative use a different type of shell. They're called that because of this type of test and how they respond to it. So the basics of how this work is you stain everything with this material called crystal violet, which is vibrant purple. And then you use mordant, which is iodine, which basically just kind of makes the dye mostly permanent. And then you use alcohol to wash away extra dye. So depending on which type it is, one of the types has a shell that's much easier to let stuff through. The other one is much thicker. So what the alcohol will do is it'll wash away all the dye from the inside of bacteria that have the thin shell, but the thick shell is too thick. It can't come back out. And so then we use a pink dye called safranin, which will just stain everything, but the purple is much more pronounced. So the only thing you'll actually see the pink on is the thin-shelled stuff that you wash the dye out of. So this is gram stain. We should have time for all of this. But yeah, that's the plan for the work, the actual hands-on EDIF. Any questions? Cool beans. Let's head over there and we will get started. Check on the live stream and see how many people are just writing butts. I suspect most of them.
Can I run up to washroom for like 2 minutes?
Yeah, go for it. Go for it.
Not very high frame rate.
You have second construction.
Here, there's a gold box out there. Just put it in there. I realize my computer is just worst for this. There we go. Going on screen. Yeah, so grab some chairs. Get comfy. We're going to be working at the silver table here.
Is there somewhere you want to be working?
So we're going to start with probably streaking just because that's the main. So I'm just going to stand at the end here. Yeah, so these are the plates that I've prepared. I'm trying to do this so everybody knows very bad practice. Never do that. The problem with plates is that they all look the same. They're all vaguely off yellow. If you don't label your plates as soon as you make them, you're in for a very bad time trying to figure out which one's which. But so these are all LB. The owners didn't label them because I knew what they all were going in. So as to actually getting some bacteria, like I said, bacteria everywhere. So I'm going to just show you how to do streaking really quickly, but then I've got these little, these are called inoculation loops. They're literally a tiny loop. So what you're going to do is you're going to go find a thing and poke it with the inoculation loop. And if I mean, it's got to get a little bit wet. So I'm going to I've got a little squirt bottle if you need. Wet it first, so that way when you poke it onto something, you're actually picking up bacteria. Because even though there is lots of bacteria on everything, you're going to do better if you actually. But then what you're going to do is you're going to go around and just poke something, bring it back, and then you're going to streak it onto a plate. I only have the one metal loop, so these are only for the very first time you touch the plate. Which I'll explain this in a second. But these are for collection. That's for actually doing the streaks. We're gonna have to take turns. But so the basic idea is
May I recommend moving the laptop closer so that we have a proper view of the table?
You can kind of see.
No photos.
That microphone.
this is the nice thing. We're just doing bacteria, right? And also because these are presumably most of the first time you're ever doing this, you're probably gonna fuck it up. So we're not really and also we're just sampling from random stuff. It doesn't matter. If we were trying to do something really fiddly, we would be working in there. But this is just for the sake of learning, so it doesn't really matter. But yes, when I do streaking, I tend to do it there. Also, you'll see that like the really seasoned professionals, like so when y'all do it, I'm gonna suggest that you flip it up like this, and take the lid off. Also, don't touch the inside of the thing. This is sterile. As soon as you touch it, stuff starts growing. So the way that like my micro professor, who's been doing micro for 40 years, she'll hold it upside down, barely crack the thing open, and like do the entire streak like this. It's very impressive to watch because she's got a stack of goddamn Petri Dishes on top of this. I know that's the like expert way. I would not suggest you try that.
Same issue with pouring in the first place, right?
Yeah, getting used to pouring these, when you first start, it's everyone taking the whole lid off and like doing it and put it back on. It's like, whereas when I do it, I do the whole stack at once and just, put down and pour. Yeah, so I need, there's a red marker around here somewhere. Okay. So, I'm gonna just, I'm gonna go grab a fresh Petri dish. So it's one of the ones that actually has heat in it.
You can go into the air. Swaps.
So most of the time the dishes you'll work with are plastic. Can't be autoclaved, by the way. They're not that kind of plastic. The way that they're sterilized initially is with copious amounts of radiation. Although you can also do it chemically. These cannot be heated. This glass can. I mean, they're not that bad, but yeah, they're exponentially more expensive, but they're really
And if you drop
so like you can like scrape out the goo and
You gotta scrape out the goo.
it is kind of annoying, but it is nice for when you're all of a sudden out of Petri dishes. You're like, oh God, what am I going to do? So you got glass. Okay, so the basics of this is you imagine dividing this into, let's go with about 3 main areas. So you're going to take your inoculation loop, once you've got some stuff on it, be it your beard or pond scum or whatever else. You take a thing, you start here and you go cross, back and forth a couple of times. Okay? And that's the only thing you're going to do with plastic one. Throw it out. Then we move on to the metal one. The metal one we can repeatedly sterilize very, very quickly, and to do that we're going to light it on fire. Because kill it with fire is the technical term. So then what we're going to do is you rotate it a little bit, and of course you're going to be doing this, so it's open, so you can actually touch the gel. It's a very light touch. You do not need to push very hard into the gel. Basically, you're basically holding the loop so it's flat and just gently dragging it across. If you poke it into the gel, you're pushing too hard. So you take the now sterile metal loop and you drag it once through the stuff that you just did, and you do the little spray. And then again, you light it on fire, make it sterile, because the idea is we're constantly diluting, right? So you want progressively less and less stuff. Light it on fire, and you drag it one more time through everything, and then okay.
Doesn't matter how many times you go back and forth.
Not really. Just try and fill the area. Don't. The mistake that everybody makes when they first start is we'll just like go. You really got to like rub it a little bit.
And just a light streak over the top of
Yeah.
the surface.
Yeah, you're only going over the surface. You don't want to dig into the agar. But yeah, so then once you've got that, you're good. There is a trick to this, wherein if you kind of tilt it at just the right angle, you can actually see the smear of where you've been. So you're able to see kind of what's going on. The last thing is when you light the thing on fire, it is obviously very hot.
So
No.
yeah, no shit. I realize. So what you want to do is you want to basically find an area that's not been used. So while it's recently been on fire, you just like touch it gently to a clean area, then do your thing. So cool it, then do it. Otherwise, you're just gonna kill everything. It's not gonna actually spread.
Is there like a protocol where usually leave it in just
Doesn't
anywhere
matter.
that wasn't streaked?
I tend to just kind of work in the direction that I'm going. So like if I'm going like if this quarter is still left, I'll use that. But it doesn't. Everyone has their own little tricks to it. It doesn't really make a difference. Yeah, so that's the basics of streaking. So the nice thing is because we're just sampling from anything, you don't need to be particularly sterile. And from the Odin, I don't expect them to be. But yeah, so everyone grab one of these. If there's not enough, I'll go grab more. So yeah, grab one and then just find something to poke. It seems interesting.
Inside your mouth, the urinal, not at the same time.
Ideally not the urinal. Try to avoid something that's aggressively gross because then you're handling pathogens and nobody's going to enjoy it. And so you want to use the itty bitty end. The big end is kind of a little much. I'm gonna go grab another.
Yeah, we're definitely gonna need one.
Yeah.
You need 2 more if you don't want one for yourself.
Okay. You bring me the UV blaster while you're in there?
Okay.
The UV glass is pretty good.
In this case, we're going to spell 10,000.
Guaranteed you smell horrendous. Now, the nice thing is, since we're just kind of doing this for the sake of learning, we don't need to worry about like wearing gloves or being clean about this. It's literally just for the sake of learning. But generally, when I'm working lab, I will wear gloves. You don't need to when you're doing this, as long as you're careful. The reason I do is because I'm a bit of a klutz. So the number of times that I've been like, holding tube of bacteria solution and not paying attention and then going onto my hands and then going, oh God.
Send out of the lab and drop the whole set of files.
Hey, those were clean. If I'm a lot more careful now if it's actually got live shit in it. That was just annoying.
Yes,
I
from all the
wasted
swearing
stuff.
I heard before.
That's wasting materials. But yeah, so I tend to wear gloves when I do this just because then if you slip or something and you poke something you're not supposed to, you could like sterilize your hands, your gloves first, usually with like rubbing alcohol. That way, if you do make a mistake, it's fine. But for today, we're not going to bother because you're just screwing random stuff. So with that, away with you. Go find the thing. If you need a little bit of water because you want to poke something really dry, I'll go grab that.
Oh my goodness. I want to do rust. I don't know why. Just to see if there's
Yeah.
Like,
So the media that I tend to use for mead is what's called a malt. So instead of LB, yeah, exactly that. It's this.
Yeah, it's what I use for mushrooms, but
Same thing. I mean, yeast is a type of fungus.
okay, I didn't know if it was particular for yeast.
But there's a chance that it might grow on LB. So yeah, go for it. Cool.
Sorry.
Oh.
Can I borrow meat and you can go with it, drop it?
Sure. Yeah. Let me I'll see what I can find. Yeah, dude. I mean, honestly, just put it in the stream. Just pull it really quick.
Can we take a?
It'll waste a little bit, but yeah, go for it.
I didn't get that.
Just so it's a little bit wet, just so that it'll actually pick stuff up. It will work if it's dry. It's not weird. You can, but sure. Not really, but I do a little water just to help pick stuff up.
Justin mentioned it. I think rust will work. I want to see if I can try and catch it.
Let's not do that because that's a pathogen.
I wouldn't
No
stop.
fun for anybody. Yeah. Try and avoid things that are very much
Just
pathogenic.
Okay.
Go for it.
Then I have no idea, so I guess I'll just
Yeah, okay,
walk
so yeah,
through
when you want to work on the thing, you can actually just take the lid off and make sure that it's facing up. So that way, you know, you don't.
Sure, I'll swab the inside of my mouth.
And then you can do your
Why not?
first little screening.
And you take out the garbage can,
Garbage is- Yeah. As far as I can tell, none of you have streaked anything particularly weird or any worse than what's already in the garbage. Yeah. So once you've done your first streak, just put the lid back on, so that way you're not letting stuff drip. Yeah. So yeah, I'm gonna show you how to do this once. So you can pass all that stuff over here.
Flip it around. I guess so.
Okay, so generally, the like, kind of where you're playing.
So like, which side am I streaking on?
There's a hair on yours.
Pick a side,
This is why you
Pick a side.
try not to move the thing.
No, no, no.
Okay, so the way this works is you take your loop, and you don't want to go into the hot part and melt it, then that's annoying. So you just go and you literally need it to go red hot. Because at that point, everything is good and dead. So it's hot, so you just touch to somewhere you haven't been, and then you drag it. So I'm holding it so I can see the little streakies, and you just drag it once across, and then do a little rubby. Touch your area, and then always, always, always torch the thing when you're done. Okay? How about it? And so it does take a little bit of getting used to of how to hold the loop. It's literally just a matter of you just got to do this enough times and you'll get used to it. But yeah, so I'll let you guys get to that. I'm going to look up the exact amount of. Actually, you know what? It says 1 teaspoon per gallon. Convenient. You don't really need a lot. That's the nice thing. So I'm going to go grab the distilled water. And then torch it.
And how this guy goes. I think it is pretty good.
Okay, just do, you did just do the first area, right? You didn't do all three?
No. No, I didn't.
Don't, always remember to put the, yeah, inside sterile, outside not sterile. So always maintain that. Yeah, careful with the really hot area or you'll melt the water.
Actually, I think I did 3 out of 4.
Try and stay near the top of the plate. Yeah.
I think I did 3 out of 4 quarters.
Okay, well, then you're just gonna end up with 1 big streak, but that's fine.
Well, I didn't do too much.
So the most important thing, or the labeling. So write the date, what type of media it is, and what you like streak from. LB. And so you want to write around the edge of the dish, because once stuff grows, you want to be able to see it there. So yeah. Also, once you're done, torch it, let it cool down for a second, and then just straight the wire back in. So that way you can hold it. There's like a particular grip, particular. Yeah.
We know when it's cold.
And then yeah, torch it again, make sure that it's sterile. And it really does have to. You've got to see it go red hot. Like yeah, there you go. Yeah. And make sure you get the whole wire, just so if you touch somewhere weird, you get fine. But yeah, just that's kind of the thing you got to get used to. Making sure that you keep the thing at the right temperature. Make sure you don't cook your bacteria, and then it's just a bit of getting used to holding the thing. And literally, the only way to do that is doing this a lot. The only reason that I'm good at it is because I took a micro class and doing this hundreds of times over the couple of months. You get very good at it, or you don't, and then you don't. But it's literally just a muscle memory thing. You just do it a couple of times and then you're fine.
Why is it that you got to do the outside of it or just?
It's a loop so that it can pick up a little bit of liquid, so that like surface tension will hold it in there. Some people just use a straight up wire, but loops are generally better. Yeah.
Will we incubate it?
So you're gonna take them home, and then you're gonna send me pictures if they grow.
Okay, who's next?
are, so as soon as you open them, not in the.
Oh, yeah, it shouldn't,
flow, but
it shouldn't be too close.
Yeah, but I mean, the nice thing with screening is that in theory you can isolate just the one colony.
Thank you, sir.
And also that's why, like, I'll do a plate just to, like, once y'all have had a chance, I'll do a plate, so y'all.
They'll show us how it's actually done.
Yeah. Once you've done this enough, because like the micro lab wasn't in a hood. You just have the flame going, so you have to do a draft. That's my traditional way to do it. Yeah, just do it, be in a room with still air, and then have a flame going, so it just forces everything up, so that stuff doesn't fall in your dish.
So you work basically under your flame?
Next
Close
to it,
it down.
next to it. Not like here, but just like here. But also, like, you'll see me, I'm gonna do it upside down, so nothing can fall into the dish. Also, one last note is that once your thing is done, always store it upside down. The reason for this is your things are alive. You're not like in a rush, but your things are alive, which means they breathe. And so part of the respiration process is to produce generally carbon dioxide and water. So think about it. If it's like this, and you're releasing water, all of the water is going to collect in a pump. And at some point, it's going to fall onto your shit and make a mess, and nobody's happy. Store it like this. That way, any water that does forms on the lid and doesn't drip into your stuff. And, you know, worst case, you can just like take it and dump it out. Which is not great protocol, but
Straighten that.
autoclave it after and you're fine. Yeah, so that's, I mean, this is the basics of screening. It's literally just a matter of doing more. And again, so try and write around the rim, because if you write in the middle, you're not gonna be able to see the bacteria if they grow.
What side do you usually write on?
Always on the gel side. Never the lid, because the idea is you want, you always want to keep it like this.
Yep.
The less of this you have to do, the better. Okay. So you write
Just for
along the
airflow
rim.
reasons, pretty much, or just things dropping out?
But so yeah, you write along the rim so that any colonies that form in the middle, you can actually see without having to like look around the writing. And sometimes they're very vague. And also once these are like growing, because they're not going to be in an incubator, it's probably going to take a little bit of time to show up. So you can move the flame closer if you need, by the way.
Will it heat that?
Yeah, the heat lamp will work. Yeah. Yeah, the incubator I use is literally a drink cooler with a ductile heat pad in it. Works really well.
Regular.
It's the kind that you can plug stuff into. So that way you can just plug regardless of the heating pad into the.
Did it come with your heating? Because I have one that came with the heating pad for my liquid.
But yeah, it's exactly that. It's a Kinetochore.
And does that make it super humid inside or?
It can. But mine's actually really dry because there's not a lot of water. But you can add a water source, but it is humid. You can control humidity. These are all sorts of things that you can try and take into account as you're doing this to help the. So for example, when I'm doing animal cell culture,
Do you want me to bring you closer?
it's fuckers. Okay. Also, they don't like a lot of oxygen. Even though we need oxygen, if you think about it, your like muscle cells or like all the other cells in your body don't actually have a lot of oxygen. They're just getting the tiny little trickle that comes from your blood. Whereas most of what they have is carbon dioxide. So in an animal incubator, you've got a constant flow of carbon dioxide to keep it at just the right amount, so that there's only just enough oxygen that they have to survive, but mostly it's carbon dioxide. Okay. The fun bit is you can use what's called the Russian method, which is you light a fucking candle in your incubator.
There you go.
And at the point when the flame burns out, because there's not enough oxygen to keep the flame going, because the perfect amount of CO2 and oxygen to keep your cells happy. Yeah, look. Yeah, you don't need it to go like white hot. Once it's red, it's good. Make sure you get the whole wire. Whole wire. Yeah. And again, try and stay out of the hot bit or your melt it.
Yeah. I'm more used to working when I'm working with a flame like this, trying to bench melt the metal. So.
Make sure to cool it down.
People like safety goggles.
Or what?
There's fire.
It's fire.
Safety goggles for this. This there's some, look, I am a big fan of lab safety, but a lot of it is egregious overkill. Like, I don't know what you've managed to do with this, that you've got something sprayed into your eyeball, but, yeah. Don't put your eyeball in fire. This is a good place to start.
Liability ruins fun.
Yeah, right? So yeah, don't hurt yourself. But otherwise, so this is what we're doing gram staining.
That's
So
really
this is a
accurate.
mixture of bacteria.
It's just bacteria.
It is bacteria. It is a probiotic with Gurgis. So if the Gurgis garbage, you add that to it. And it's good dirt, because what makes good soil is the organisms that are living. This is all the good work.
I've been doing that with
Yes, yeah, worms are, like, vermiculture is a great way to do that. Yeah. I'll get you parafilm for that.
Yeah,
This is all
yeah,
good.
I know. You didn't have it out, and I didn't want to worry.
Yeah, I'll go grab some parafilm. This we're gonna use for when we actually need it.
Parafilm's like that waxy stuff, right?
Yeah, parafilm it's basically like cling wrap, but science grade. Also, you're not a real scientist until you wrap your life in parafilm. And dry your tears with Kinetochore.
Yellow.
No, I didn't go that far. On my face.
and then I think is there else?
Anyone else?
I think yeah, just is it cool? Just bend it back.
Okay.
Yeah.
Yeah, I'm gonna do a streak really quick, but
Oh, sorry.
But it's fine. It's fire. In the little one. Brand new roll of parafilm.
As nice as the drawing is, I would do
Parafilm
recommend
is
it
basically
as kind of obscures
the moisture in. Because the other thing is if you just leave these sitting out, the gel will actually dry up.
The gel's making sugar.
Yeah, basically. So yeah, it's a special sugar, but it's basically long. It's closer to like cellulose more than actually. Yeah, this is parafilm. It is amazing. It's all that you know about parafilm. But so the nice thing with it is that it's stretchy. So it comes like this. It's like a waxy substance, but pull it, extend, and then when you do that, it also stick to itself. Yeah, sometimes it'll make a little hole, but that's the nice part. It's extremely long. So you can just stretch tiny little pieces way out, and then you can do this where you can cover everything, and it's like, oh no, it ripped, but it sticks to itself, so who cares? Or that out. Okay.
2.5 squares.
2.5 squares.
That's exactly the MI. Yeah, you can do it on two.
You're very, very careful. Okay, so I'm gonna do one streak, and then I'm gonna need a volunteer to make some media, and then we're gonna do a gram stain. So, parafilm for anyone who wants to wrap up their dish. It's really not strictly necessary. The only time I ever actually parafilm a dish is if I'm storing it, and once these are made, they're sterile, so they'll last as long as the liquid remains. So if I'm putting like a stack of dishes in the fridge, I'll parafilm them to just make sure that all the moisture stays in and doesn't just dry out, because then otherwise you'll end up with like a film of agar and very sad Petri dishes.
So it's sterile, even though we
Kind of sterile.
Well, it's not really sterile anymore, but this one is, because there's nothing on it. So, leave on fire. Okay, I'm gonna streak from this stuff, which is what we're actually gonna do the gram stain on. So as I was just saying, this is a soil probiotic. So it's basically just a mixture of different bacteria that are all really, really great for soil. Like some of them will fix nitrogen. So like, if your soil is low in nitrogen, rather than adding fertilizer, you add bacteria, and the bacteria will just make more fertilizer from the air. Which is, yeah, really, really quite. Yeah, also known as it's just goddamn bacteria. So there you go.
nanotechnology, aka bacteria.
Little drop button.
Check out the master.
The nice thing is I don't need to cool it down because I'm sticking it in the steel. They can't go over. Cool. So I can see where my streak is. So I know where I'm going. Okay, I'm gonna go that way. Pop it up. Off. Fire again. And, people will say that, everyone's got their preference on exactly how to do this. Doesn't really matter. Whatever you get comfortable with. Yeah. Little thing. Again, you just pull it through the area you've been in. And when you're done, let it fuck around. Things will never hear Professor say. Place it in. Marker, please. B. A.
Oh wait, we're supposed to write out.
Yep. You gotta write what kind of media it is. Agar is the delegate. It'll help you. You go by the food. Everything has agar.
Then I can write out.
It is
Taking
voila. So in my case, I'm gonna put this in the there.
And I should write out.
But yeah, that's the basics of streaking. Like I've said about 1,000 times at this point, just do it just more often and you'll get used to it. And especially once you're working, it's really stretchy. You only need you really only need you don't need to try and put it back. Just rip off 2 squares and call it there.
Only 2.
That's pretty much all you'll need. If there's extra to share. But yeah, once you get working in the biolab, you do this a lot. If you're working, but this is for basically all microorganisms you do this. If you're working on animal cells, it's different. The way you handle them is different. The way they interact is different. The food you give them is different. You never use agar. They're always in just liquids. And instead of Petri dishes, they're almost always in flasks, which are like flat, rectangular looking things. These animal cells, rather than growing as colonies like this, will actually just fill the dish and coat the entire bottom. But that's a whole thing that is well beyond what's being covered today. Mostly because we don't have any of this stuff for it. But it's not actually that difficult. Because it's all liquids, you never need to do streaking. It's nice. You literally can just suck up your liquids. There's more steps, but the catharsis of it is I particularly enjoy.
That'd be for like mammalian.
Yeah, all your mammalian cells have their own tips and tricks and stuff.
They have different plates too, right?
Yeah, that's the plastic. So instead of doing this, when you're moving them around, you use serological pipettes, which are these big, individually packaged 5 or 10 ml pipettes, and you use an autopipetter, which is like my favorite thing ever, because you just pull the trigger and it pipettes. And you're like, oh, okay, we're done, and then you just pick it up, go to the next thing, eject it, on and on and on.
On a big laminar flow.
Yeah, and then you gotta work in the big fancy hood, and you have all the noise of it. So you just throw in tubes, you just moving flasks. It's great. You're like, I need to make 16 flasks of Chinese hamster ovary. Okay, here we go. They're just called CHO cells. But yeah, so the types of cells that we were working with, we had fibroblasts, which are skin cells. We had CHO, which are Chinese hamster ovary. We had HEC, which is human embryonic kidney. All this different stuff. So depending on what you're doing, you do it on different stuff. They all grow at different speeds. So with this, at these temperatures, it's going to take a couple of days to grow. a week, it could take more. In the incubator, I'd see growth in a day or two. Whereas with like fibroblasts, once you've got them growing, once you put them in a dish, you got like 12 hours till those fuckers have filled the dish. Like they grow real fast. So if you don't get your timing right, your ass is in there at 4 in the morning, like plating your cells. Because if they fill the flask, they die. So they freak out. They literally freak out if there's too many of them. So your schedule is set by the cells. So if you know what you're doing, you'll like do that as the last thing you do at the end of the day, and the first thing you do at the beginning of the day. And then once you have enough, you basically just get them all off the dish, pellet them down using a centrifuge, and then basically mix in glycerin, and then freeze the fuckers in nitrogen. And then they just stay there until you need them again. And then when you need them, you take them out, get them growing again. But yeah, it's like a whole thing. I particularly love it. The catharsis of it is amazing. Whereas this is more fun. But very different labs. Mammalian is like 50K to get that lab going, whereas this is like a couple of K. Yeah, so that's that. So we're gonna make a little bit of media. And you volunteer. Okay, cool. So this is my box of different types of food for the various organisms. So I have, like a kilogram of lactose. But so what we're going to be doing is we're going to be using this stuff, which is yeast nutrients, which is basically just urea and ammonium phosphate. I'm contemplating whether or not I want to throw a little bit of like salt or something in there, but I don't think we're going to bother. We're just going to see what happens. Yeah, so this says 1 teaspoon per gallon. So I don't know what a teaspoon is. 2. Yeah, I know, seriously. Why do things come in freedom units? This is for not freedom. But yeah, okay, so 1 teaspoon per gallon, we're gonna make, I don't know, 400 mils. So how many? 3.84. Okay, 3.8.
I'm sure one of us could just add the searing bill.
I reduced. Yeah. I have. So it's 3.8. 3.8 per gallon. That's not a lot of teaspoon.
None
And
of
they
it.
quit.
Dividing a teaspoon, but you don't know how much a teaspoon weighs.
Yeah, I know. No, but this is how much of a teaspoon you would need for 400 mils. Not a lot. We're gonna go with a gram. A gram sounds nice. Let's go with a gram. If it doesn't work, who cares? It's right. I like a gram. We're going with a gram. So here's the scale,
It's roughly a quarter teaspoon.
which has everyone on the internet commenting, oh, it's a drug scale. Well, good enough for them. It's good enough for science. Okay, so there's scale. So grab a little piece of aluminum foil.
Scale actually a lot larger
You
than
just want like a piece about the size of this. It's just something, it's called a weigh boat. It's just something to weigh out the stuff on so you don't make a mess of the scale. Because it's not drugs.
Sorry, what are they called again?
What?
Weigh boats?
Weigh boats.
Okay.
Yeah.
Weigh boats.
Well, you can buy the free,
The plastic one.
Yeah, you can, yeah, it's literally what it is. We're good, we don't need more. So the trick to doing this is once you've got it set up, make it a little divot in it. So that way, once you pick it up, it's easy to pour it into the flask. So normally, when you're making media, you use this stuff, this is the actual LV. So you would use this and agar, which I don't have out because I'm not making plates. So in this case, when I made the plates that y'all just used, I made 200 mils of media. So I weighed out 4 grams of this and 4 grams of agar, put it in the flask, add 200 mils of water, give it a good shake. And then what we're going to do, so here's your, where did I put it back in? There you go. So turn the scale on. Put that over a hair, so you can see what's going on. And then click tear. So tear basically sets the 0. So now, you just do it anyways, it's just good. So this is a scoop. So you scoop, way underground. The scale's kind of finicky, so work fast or it will turn on. Yeah, and if it, like I said, do work quick. You're gonna want to, yeah, there you go. Yep, there we go. We're just doing the one. Okay, cool. So now we got the one in there. Pour it into a glass. Cool. Now I need another volunteer. Sure. Steve excited? Take some water. Let's go 400 mils. Put the eyeball on this because this is the first test, so we're just gonna see what happens. So there's gradations on the side. Generally, you would use a graduated cylinder for this because it's way more accurate. But since we're just going with YOLO swag, just use the thing on the side. Yeah, this is learning how to pour liquids is one of your like prime lab skills. It's generally best to not rest the thing on the thing, but since you're just getting started,
I'm actually not. If you look, my hand's under it, it's slightly hovering. I'm using it more to make sure that I'm
chemistry pour. You've either been bartending or doing a lot of chemistry. It's fine if you went over. Okay, so then the last thing that we do before this can go into the autoclave is, I mean, obviously we got your remote mix. It will dissolve because it's about to get the shit heated out of it. But you want to give it a little mix so like it mostly dissolves. This is actually a fairly large amount of liquid to go in the autoclave. This is about the most you ever want to do in a flask this size. But because there's no gelling agent in here, it's not going to foam. If you were making like the plates, the media for that, you'd only ever do like 200-300 mils of pops because it will foam when it gets hot and it'll come spurting at the top and make a goddamn mess. So you do little bits, then if it foams, okay. But this, because it's basically just salty water, it's fine. Honestly, for what we're doing, it probably doesn't even matter if it's sterile, but we're going to do it anyways just so you see the processes. So once you've made your thing, be it LB, this, whatever your recipe calls for, just making a cake. Take some tin foil, give it a tin foil hat.
We're all scared of the government.
Yeah, glass is terrified of the government and contamination, so we give it a tin foil hat. And then we use this stuff, which is magical. It's autoclave tape. So there's actually little lines on it that will change color if this has been sufficiently sterilized. So if it comes up to temperature, it turns black. Not black when you pull it out, you haven't cooked it long enough. Take a little piece of autoclave tape, put it on the tinfoil hat. That's basically it. So no, so the nice thing with an autoclave is it gets really high pressures. So you're actually getting way higher than the normal boiling point of water because everything under that really high pressure. So it makes sure things go really good and dead. So yeah, this particular autoclave, every autoclave has its own little quirks, but this is the style that you're probably going to interact with most unless you're just using a regular pressure cooker, which also works, but it's not quite as good. I think you get to a higher pressure than a pressure cooker can. Pressure cooker will work, you just got to run it long. I like it quick. So yeah, take the lid off. There's already water in here. But even then, you always want to check because if this thing runs out of water, it gets a lot hotter. Your shit melts. Okay. Or burns. A quick little check. So yeah, the water level looks okay, not that cool. So I think I'm actually gonna go grab my water container and just pop it up a little bit. You can actually have a look under the little main buckety bed. There's little feet. You pick up the bucket so you can see under. So whenever you fill these, you always want to use the you always want to use the actually can you that out? You always want to use the cleanest water you can because if there's any salt in there, it will build up. and then cleaning is pain in the ass. So I'm using just nice rainwater. So you basically just want to go to the bottom where those feet are. We're like halfway up the feet. That way you're guaranteed to have enough in here that's not going to run out. And then literally all you do is take your thing, whatever is being sterilized, load it into the top. Not in the top. There's a drain in here so that if any like this fills up, it'll drain out. There's also a little platform fit to keep this off of any water that drips in there. Just back. Make it massive things. To get really hot and to add pressure. So instead of having to like get an air compressor and manually compress this, you can just evaporate a lot of water whilst contained and it'll make pressure and stuff. Then you take this and you gotta make sure that there's like a little tube in here where this fits into. This is the actual pressure gauge. So take it, put the thing in the little tubey and then line these up. Whenever you're doing this, you always do the opposite one at the same time. Because if you try and go clockwise, you're going to put the pressure unevenly and the thing goes up and then back. I don't know if you've seen that Mythbusters episode where they blew up a water tank.
Oh, yeah, that was interesting.
Yeah, picture that, but big ass metal thing comes flying at you. No good.
Wasn't that big ass mountain that exploded?
Yeah. Just bigger. If you look up exploded autoclaves or pressure cookers, you'll see like lids of them embedded in someone's ceiling. It's pretty fucking gnarly. But yeah, so you just line everything up best you can. And then you just go to finger tight. You're not like wrenching this down. It will add more pressure as it expands. There's a gasket here that is like leaking. But so then you just kind of go in a clockwise motion. Tighten this one, tighten this one, tighten this one. And once you've done that one, you just root check everything because it'll loosen one of the other ones. Yeah, okay, that's good. That one's good. That's good. And then you plug it in. Plug it in. Plug it in. They're not wired in. That's it. Now we just wait. So this is going to take a while to come up to pressure. When it does, this thing is going to make up for that noise. Before then, it's going to make some really annoying, like hissy noise. And then all of a sudden, it's going to pop and release a bunch of pressure. Generally, the way that I do it is once it does that, I'll set a 10-minute timer. And then it'll be at pressure for 10 minutes. Get it down. Which you just unplug it and then don't touch it. It'll cool down and the pressure will go down. If you try and vent it before, like if you're in a rush, you can. But any water vapor that's around will suddenly come out of like gas phase. So if you got anything in here that's dry, there's any water vapor in there, all of a sudden it's going to fresh. So whenever I'm doing like pet tips, never vent this in advance where you're going to make a goddamn mess. And my bed box is like half full of water, which it's sterile water, but it's still in. You don't want that. But yeah, it's basically it. So there's a boiler in the bottom that's literally just going to heat this up. Just like any, kettle, basically. It's a fancy kettle. Like a club. So, it's a good kind. But yeah, that's basically it. So once this is done, just let it cool down, take the thing out, and then do whatever it is you want to do with it. If there was LD in here, so like with agar in it, you've got to take it out before it's cool. Otherwise, it will solidify in the flask, which is no good. So with LB, there's this sweet spot of just cold enough you can handle it, but still warm enough that it's liquid. So at that exact point, you can actually pour it in plates and let it solidify. Cool, make sense? Okay, good shit. Okay, we're gonna do the last little bit, which is the gram staining, and then we were done. So for that, these are, this is all the gram staining stuff. Comes in a wonderful little kit in drop bottles. For this, we are actually going to work. I'm going to volunteer for this one. Cool. So the way that we're going to do this is actually fairly straightforward. So I've got microscope slides over there. There's a little box. Goes this comes into. There we go.
Little box, not that one.
Yeah, the not so little box. Okay, so microscope slides. It's basically also but in class. Not like them. Anything else to do? Not really, but So these ones, for whatever reason, microscope slides tend to be fairly cheap. They have this annoying habit of like sticking to each other in really weird ways. I'm actually gonna see if I can find one that's not stuck. Because when they're stuck, it's usually because there's a little bit of oil or something pulling it together. Yeah, there's a little bit of debris on it, so that's fine. So basically what we're gonna do is first we need some bacteria to stain. So there's sort of a trick to this. And I'm gonna have to go grab a pair of pliers to be able to do this. But I'll be right back.
Next time, will he find the pair of pliers?
Well, no.
I organized the whole place and know where they are. Pliers. The least scary time you need pliers with biology. So get the plane going again. Okay, so this is what we're gonna grab stain. So what you want to do? Okay. So just get a drop or two. You really don't need a lot. Literally just a couple of drops onto the slide. And try and hold the slide flat so it doesn't run everywhere. Yeah, perfect. So just a couple of little drops. Perfect. Yeah, spread it out a little bit. Up and down. There we go. Perfect. There we go. Yeah. Cool. So now what we need to do is we need to dry this. Now, slow way to fast way. Ideally, you would just like let this dry so we have kind of a smear. But for the sake of this, try and do this the fast way. This might fuck it up, but we'll find out. So, yeah. It very quickly dries. The trick is you kind of want to.
Not leave it there too long.
Yeah, I'm just trying to get a little bit of the stuff to evaporate because ideally what you want is you want to let this dry all the way because if there's still water on here, you'll pop all the bacteria up. So I'm just trying to just heat it up very gently, just to. I don't want to go all the way to dryness, but I do want to not have this take all the way. Almost dry. Oh, kind of nice. The reason is we really want this to be.
I think it might sound.
Yeah, basically the reason is we just want this to actually be properly dry, because if it's not, then once we add the other stuff, it's all just going to come off. A couple little passes. And there's a chance that this might be totally fine, so I'll try it again. This is sort of the thing with biology. The tray around here, I think it's a pattern. Like a little tray, what do you think? Yeah, there we go. Cool. So, yeah, you know what? There's a lot of still kind of warm. So we're basically letting it cool down. There's also the other thing that might have happened is try and get those back.
Yeah. Also try not to cut yourself.
Yeah, if any of those broke, check them out. But also be really careful because they're glass. So yeah, the thing with this is it might also be too concentrated because it's straight out of the bottle. So sometimes you end up having to try this a couple of times to get it to actually work. But you know, within reason, you're generally, you know, who cares? Like the reagents are very, very cheap. You only need a drop or two of them. So if it doesn't work the first time, it's fine. Dilute it a little bit, try again. So all we're gonna do is I'm gonna use a couple of these to pull some of those out. The reason we're using the tray is because we got to drip stuff onto it, and the reagents are not particularly pleasant, or do you want to get them on your hands? So whoever does this is going to have to wear gloves. Okay, there's our little dyeing tray, super improvised. But you can't see, we're done with this now. Yeah, if you can't see, then do come here so you can. So these are the gram staining reagents. So just to review, first we do crystal violet. Stain everything purple. Then we hit it with iodine to make sure the dye is permanent. Then we do ethanol to wash away any dye from the thinner bacteria. And then we do safranin to make everything else pink. Cool. There's also sort of a sweet spot with doing this. Like that graphic looks really nice, but there is a sweet spot to this. It does take a little bit of doing to get right, because if you use too much ethanol, even the thicker stuff will get washed away. So there's sort of like a sweet spot of just the right amount will get it to work properly. And if it doesn't, it's not a big deal. But the point is that y'all get the idea. So, all here? Tell me about it. Okay. Grab some gloves, whatever size you are. Yeah, we're just gloving up just because we really don't want to get this stuff on your hands. It won't hurt you, but you're going to be purple for a while.
Not easy being very usually more powerful.
Yeah, I mean, Google's iodine is basically the same stuff you get at like the pharmacy. Like pure iodine, it's like it's just safranin. Ethanol, which you tell it's the ethanol because there's no writing left on the sticker.
Yeah, it's such a little. It melts plastic on the side of the
Just a little bit. There we go. Freedom. Okay. So what you're gonna do is you're gonna start with the crystal violet, and you're just gonna put a couple of drops onto the little smear, and then basically just tip the slide back forth with it. Spread it. Okay. And then once everything's good and covered, like let it sit for a couple of seconds, and then we're gonna pour it off. And so this is also part of the
One or two drops?
Just a couple. Yeah, it doesn't need a lot. Stuff is very concentrated. Yeah, so then just kind of tipper back and forth to distribute it. And if it's not distributing, you can just use a pipette to just spread it around. And then just use the water right over here to just rinse her off. But let's just let it sit for a couple of seconds. Just to give it a little bit of time for the dye to sink in.
Should we add it more or?
So the dye is actually sinking. So.
Yeah. But yeah, so then just give her just a really gentle rinse, because we don't want to like
Squirt everything out?
Yeah, we don't want to like rinse it all away, because we do need to do the iodine, but like just a really quick rinse.
Okay, do I like tilt anything?
Yeah, tip, and then just a super gentle rinse, and do it into the tray so that anything that comes off. Just another couple of drops, just to get the main at the end off. So just rinse off the edge, basically. So like tip and let it run off. There we go. Okay, now we're gonna do the iodine, which should be this one. There's a fair chance that the timing of all of this isn't perfect, and the exact amount of bacteria on the thing isn't perfect. Like when we did this in my class, everybody ended up having to do like 5 of these before it actually worked properly. You end up fiddling with it to get it perfect. And, like, you let your smear dry naturally instead of torching it so that it doesn't explode at all, and all it is just didn't. So yeah, no fun in success. Something like that.
How many drops?
Same, just a couple of drops.
Just repeating the same.
Exactly, same concept. And then this one we're gonna let sit for, like 30 seconds.
Spread it around.
Yep. And the nice thing is everything's a little bit wet already, so it should spread a little easier. So again, this is the mordant. This basically just tries to make sure that any of the crystal violet, the purple stuff that is in the cells is going to stay in the cells, or at least better than they otherwise would. Yeah, so we give this a couple of seconds, and then once it's done, we'll rinse it off again, and then we'll hit it with the ethanol.
Are you using too much water, or should it just be like drops of water?
Yeah, it's
More controlled.
it's literally you just putz about with it a couple of times, and eventually you'll get it. Sometimes you gotta use a little more, sometimes you gotta use a little less. But
Why did you try it? It's just not like
because you need the bacteria to be stuck to the slide. If it's wet, they'll just wash away as soon as you do this.
Right.
And then the like torching them basically like kind of permanently adheres them to the cells.
even though they're used for
Yeah, exactly. And so just dump it, give it a really quick rinse. And really try and rinse all that stuff off best you can. Or just a little bit. And try not to shoot directly at the smear. You always want to try and aim off of the smear, so let it run over.
Oh, okay, gotcha.
Yeah, so hopefully it works. First time I've used this particular thing. So it's the same thing. Anytime you buy any of this, yeah, perfect. So I'm gonna grab a little bit of paper towels so you can dry that off. Anytime you buy one of these kits, always gotta end up fussing about with it a little bit, just to get it right. So yeah, just dry off the underside of the slide.
Underside?
Yeah. There you go. And then just kind of like dab the top corner, just try and get as much of that off as you can. So that's perfect. Okay, now we're going to do the decolorizing, and so you can see that there's actually purple stuck to the slide. You just pull it up so you can. So yeah, it is actually purple. So that means something got stained. Good. So now we're going to try and remove just the right amount, so that some of it stays, but just the kind of bacteria we want, basically. And this is sort of this is usually the fiddliest part. Number of times you've either over decolorized or under decolorized, so either everything's purple or everything's pink, which is not great. So, this is the ethanol. So again, just a couple of drops. Yeah, just a couple of drops. And then this one, we really don't want to let it sit too long. So like, 20 seconds kind of thing.
1 or 2 drops.
Yep.
Everything came right out.
Yeah, okay. And so number, and give her a rinse.
Like almost everything came out.
Yep. There only needs to be a little bit of like a hair. So this is the saccharin, so that's the last one. So again, put it on a couple of drops, give it 30 seconds, and then dump it, rinse it, and then we're gonna basically let it dry, and then look at it under a microscope. See if we got it. Yes, please do. Please do. It's not that I'm worried about it drying so much as somebody knocking it or being purple for a week. And crystal violet, not super great for you.
No?
It here does wonders on the skin.
Yeah, makes your ass purple.
Yeah. It's just fast. Wonders.
Yeah. That's the real secret of the Petri Dishes. You just take crystal violet baths. That's why you put it. Cool. Yeah, so let her sit for a sec, and then give her a rinse, and then we're ready to have a look at it. Although we're going to let it dry first, just because, yeah, rather not get that crap all over my microscope.
That's not the modded one, eh?
It is, I took the bottom. All right, take a rinse.
You have any sort of your violet?
Wait, what?
You have a heater and violet or anything? You can take over here. Let's take like right.
Not really, but there are things that get hot in there, so it doesn't get too cold. But also, the reason it's cold is because it's a weekend, and the dude who runs the building is kind of cheap. So during the week, it's much warmer. Like you actually turn it on. But over the weekend, it just like slowly gets colder. Which is one of the reasons why I built the incubator, because otherwise stuff just never would grow. It would just take ages and ages and ages. Whereas now, you know, I'll have a culture ready in a day or two. Yeah. Pat it dry, and then just kind of dry the top bit away from this, like in areas where the smear isn't. Basically.
So like that.
Yeah, so like try and hold it so that everything blends to the corner, and you can just kind of dab the corner, and it should collect most of the liquid. And then we can just kind of wait for it to dry. So look at it. Okay, see a little bit of purple, a little bit of pink. So hopefully we got something. But we'll find out in a moment. So just, yeah, leave her down and let's just let it dry. Yeah, that's basically it. The only difference at this point is just fine tuning this, the amount of water you use, the number of times you wash it, all that kind of stuff. Yeah, basically it.
And you're pretty much just letting the water kind of just wash over it. You're not going directly on it.
If you shoot it directly, then you will actually dislodge the bacteria. So it's
You're gonna keep it on.
You also probably dislodge some bacteria. Yeah. But that's like, it's all the process. That's it.
Which in this case is actually kind of a good thing. There's way too much in that bottle to go from. So rinsing off a little bit extra is probably will have that part of it. Yeah, so we're basically just gonna hang out until that dries. Then once it is, we'll look at the microscope and see what we see. So that being the case, I'm gonna take like 2 seconds to get the microscope set up. While I do that, are there any questions, comments, thoughts?
Off-topic question?
Yeah, sure.
Was one air filtration unit never done?
Oh yeah, I mean, it's a huge unit.
Yeah.
It's enough that I actually had to turn it down, because otherwise the whole room would. Yeah.
Because you're saying you might want to put another one in there?
Yeah. We do not
Not
need
at all?
another one. We'll have that all. Yeah, so. Messing with this thing, trying to turn it into a fluorescent scope, which did not work.
So you have a fluoride and filtration through your
No, I don't have flow hood. I just filter all the air. So the entire room is the flow hood, basically. So that way I can just work. It's not perfect, but once y'all leave, because I've been going in and out, I would turn UV on and just torch everything. Okay. So that the UV will do a pretty good job of getting rid of most of it. Say it, whatever. No. This is much deeper UV. So that UV is around like 350 to 400 nanometers, whereas that UV is around 250. It really rips things apart like a lot. So
Leave it on all night kind of thing?
no, 20 minutes.
Yeah.
Yeah, 20 minutes and you're good. And it also produces an enormous amount of ozone because it's got enough energy to rip air apart and shove it back together. So the ozone will do a good job of torching everything a little bit.
That was like a special weird.
Yeah, I mean, the thing is, that entire, eBay. Mostly, I'm not in the States. If I was in the States, then yes, but.
Does Amazon suck that much per year? Yeah.
Wow. Oh, yeah. So I order from eBay because then I'm just getting directly from China, basically. Okay. Whereas in the States, you get everything from Amazon and it shows up in a day on Prime, which is amazing. Whereas here it's just absolute fucking nightmare. Yeah, but you know, it makes sense in other areas.
Yeah, maybe too.
Yeah. So cool. How about
pretty moist.
pretty moist? Okay. That's a different topic. Wrong book. You don't love glue for the bacteria. Reasons I like doing these workshops. I can say things that would get a professor fired. Oh, these are.
Have you done workshops?
Not here, but at the place in Florida. We did basically this.
Did you get farther? Or
No, less hard. I got way further today than I did that time, but that was because we were only doing like street basically. Talking about it. But yeah. This is also, I mean, we've taught like an entire bio course. So I didn't like rehearse that. This is just like I already know it. So I just like Like, threw up some pictures on slides. I'm like,
Where'd you teach it?
in Chile and Brazil. That was weird.
Speak English enough?
Yeah, well, I mean, it was mostly like rich white people who could afford to go down to Chile for a random biology course. But it was interesting nonetheless. That's okay. I just want to make sure there's nothing on the bottom. That's really the thing that's gonna make a mess.
Just cleaning out underneath.
Let's get it around. So the way that we did this back in my class was you just put oil directly on here. But I'd rather not get bacteria all over my lens. So I'm gonna use drops. So like put it down.
Why wouldn't you? Okay.
But then you risk taking that stuff. The thing is, bacteria are very, very tiny. So I'm not actually expecting to see much at replication, but if you want to get it dialed in.
Purple?
Think about purple. I'm trying to find a spot that's a better mix. Where is it? One second, but just trying to get this dialed in. Because microscopy is one of those things that take a whole lab class to teach you how to do it properly. I'm taking second class, there was one girl who just could not figure it out. So she spent the entire, or the whole thing that we were doing was find the thing of the pre-made, slides. So you can, draw a little picture of it. Took her 2 hours, find nothing.
Bless her heart.
Yeah, bless her heart. Then one of us was like, hey, fuck this, let me try it. Took us 30 seconds. We're like, okay, dude, you were going the wrong way. Right. What do you know? Remove more mods from the microscope. Yeah, gotta remember to unmod it. Okay, well, I can see some stuff. Okay, well, if you guys want to look, you're welcome to do so. Things look mostly purple. But I suspect that if we were to hunt around, we'd be able to see it better. Also, I'm not at the maximum magnification, so if I were to go all the way down, we'd see it a little better. But I just want everyone to have a look first, just so that you can see what it's like. And then once everyone's had a look, I'll go down all the way once I've found a good spot, basically.
What was it? Gram positive or purple, gram negative, pink?
I'll be honest, I can never remember.
Okay.
I always get that backwards. I feel like gram positive is purple. So that's what we're looking. No, wait. No, gram positive are lactobacilli, which are.
Although it looks like some liquid starting to cover it.
Was it? Oh, okay. Well, I don't know, man. I very rarely have to do a gram stain because I almost always know exactly the thing I'm working with. Like this is like this whole process is something you only really do in micro. If you're
As a lab and not a practical person?
no, I mean it is practical. Like it's if you're trying to identify something from a sample. But that's very rarely a thing that I personally do.
Yeah.
So it's one of those things. It's good to know how it works. Good to know. It's good to do it once or twice.
What's one case where you would want to use it?
Like if you're trying to figure out what's in that bottle.
Yeah.
Like you want to see an idea.
So it's more just identification purposes.
Exactly.
So if you already know what you're dealing with.
And that's the thing. I do genetics. I don't do micro.
Yeah.
So I always know exactly the thing I'm working with.
Right.
Just because the alternative is obnoxious.
So if you were scouting around in Amazon, Probably not
Exactly.
like you put on your backyard.
Exactly, right? Like if you're just like swapping your compost. And then there's
Can it
other
give you
things
like a
that we look at.
ratio breakdown of things that are in your soil?
It can, but it also depends how you handle it. Because one of the biggest issues that we have in microbiology is that it's really shite for growing most organisms.
Yeah.
Because when you make LB, there is phosphates in the LB just as a function of the fact that you just exploded yeast to make your food. And so when you autoclave phosphates, they react with all of the other stuff in there to make highly reactive species. Species being species compounds, not bacteria. And that'll actually just that will prevent most bacteria from growing. So the way that we figured out how to grow, and also the other thing is most soil bacteria are extremely dependent on the other bacteria around them and refuse to grow if there's not all of their friends, basically. They are the whiniest 3-year-old.
They're social creatures.
Extremely. But more than that, like a lot of the times the other organisms are making things that they require to survive. Because they don't make certain amino acids, for example, so there's a lot of sharing. So basically what they figured out how to do is they took a hunk of agar and just stuck it on some dirt and just waited. So that way some will stick to the plate and start growing with the help of all of their friends. Or they'll do it with like a membrane. So they'll take like a drop of like dirt on the top of the membrane and put the bottom of the membrane onto the like wet dirt so that the molecules can diffuse through, but the other bacteria can't. So that way you're able to find way more delicate organisms. And I mean, from that, we found whole new classes of antibiotics.
Yes.
Because now we're unlocking these huge new areas of the bacterial world because now you can deal with the fiddly ones. And generally, once they're growing, they're okay. But getting them growing that first time is hard. Also, one of the experiments that we did when I was in school was we basically made LB and dilute LB, so there's less actual food in it. And there was a huge difference in the amount of stuff that would grow. Like full-on LB, 2 or 3 different organisms would take over the entire plate very quickly. Whereas the dilute LB, there was hundreds of individual colonies that were all different. Why? Too much food, overwhelmed them.
Yeah.
They were really delicate. They were really, really finicky.
So it's just like environmental factors.
Exactly.
Temperature.
They're just fiddly little things. They like to be in their little niche and they don't like to be fucked with. Which is one of the reasons why, like the way that we farm, for example, when we're constantly dumping shite onto the soil, is really not conducive to all of the organisms living there.
Right.
Because then they become dependent on the fertilizer function, and all of that like nice interplay is totally fun. Which is one of the reasons why you would add a big bottle of that to your soil to help replenish that, because you've just nuked it.
Would you also like maybe want to look at like what's actually in the soil and see like something that's growing naturally before you just dump random assortments?
Yeah, the stuff that's in there is one of the best collections. So one of the bacteria specifically, the reason I bought that bottle, because I'm trying to isolate one particular bacteria, called R. palustris. It is known as the most versatile bacteria ever described. It can eat anything, and I do mean fucking anything. It's photosynthetic, it fixes nitrogen, it fixes carbon dioxide. So does all of the
What do you mean
things.
by fixes?
It'll take nitrogen gas and turn it into protein.
Oh, really?
Yeah, or nitrates at the very least. It's very similar to the purple bacteria that live in the roots of beans. Because the reason that you plant beans is because they have a symbiotic relationship with red, purpley bacteria in the soil. So they live in and amongst the roots in form of little nodules that replenish the soil of all the nitrates. This is why traditional farmers know that you've got to plant things in a particular order so that the soil has time to be regenerated by these organisms. So the R. palustris not only can it eat everything, it shits electricity if you cut off its oxygen supply. So it does everything. Yeah, it can also eat acrylane, which is one of the nastiest compounds we have ever cooked up. It's like the precursor to making acrylic. It is horrifying, like unbelievably
So
toxic.
is this relating to your super?
I want to upgrade that bottle, and I want to make a bucket where you can put like plastics, solvent, like any nasty shit you can come up with, and it just turns into compost. That's the end goal. Yeah. So the idea is,
Oh, just more of that, or?
just get dirt and thing. Good.
Dirt.
So you just want to isolate the bacteria?
I do, because I want to work with them specifically. I would love to use them instead of like coli.
You're gonna fill them.
Yeah, I would love to be able to do genetic mods with them, do sequencing, do all kinds of interesting stuff.
Because they're more.
They're extremely versatile. They'll eat anything. Like,
I'll see coli will get out.
And I wish I eat anything.
yeah, and also E. coli are gross. They reek. They smell so goddamn bad. Like,
And if they get
and
any.
my delicate sensibilities do not like fucking E. coli. So I know a guy who grows 55-gallon drums of E. coli at once, as a DNA production factory. So they are the most efficient way of making shitloads of DNA. It also smells like death. Because unsurprisingly, the bacteria that lives in your pooper smells like your pooper. It's not good. So it's a very particular, just awful smell that I personally cannot stand. So anything I can do to gently get away from that nonsense, the better. But yeah, so and also the whole they make electricity thing. So being able to throw them into a microbial fuel cell and just you feed them literal shit to make electricity sounds pretty cool.
If you could, I mean, how would you just eat?
No, they
No,
literally
just cut
shit
the
electrons.
So like they're conductive or?
No, they literally like release electrons as part of their when they don't have oxygen. It's just part of the way that they do. So you stick an electrode down there, and you have a second electrode at oxygen, and so it basically forms like an artificial respiration system.
So it's
Some
sort of like
forms
a potato battery.
Yeah, it's like a potato battery, but it's eating shit.
Not just
Yeah.
shit though, just like
Anything you throw at it, which is one of the reasons why microbial cells are so cool.
So would you take some of the superworm bacteria genes and maybe put it in that?
Maybe that's one of the reasons I want to isolate them is because I do want to figure out what lets them do that.
Yeah,
Because once I know what the enzyme is, I can just clone it out and stick it to something else, call it a day.
that's gonna be the most interesting thing I could possibly imagine, yeah.
Yeah, we've got one on order that'll break down PET. Yeah, because we already know the enzyme that does that. And my actually it's an E. coli with a little piece of DNA that codes for that enzyme in it. So I'm gonna just take that DNA out, stick it to something else. Add gene.
Store for genetic.
Yeah, and they ship them in E. coli. So that's
Listen,
why they
in
ship.
the internet, there's a store for everything.
Yeah, there really is. But with AdGene, you've got to be either a nonprofit or academic to order from them. It is. But you've got to be a registered lab. So I just order through the Florida lab and then they can ship it. Yeah, so that's basically it. I'm gonna poke at this really quick, and then we're basically done. That is all of the things. I do hope you're using money with no special because I think I've covered as much as I physically can. That is my zoom right in and see what we got. So, anyone who's never used a microscope before, where did they go? Sure, then.
Just like a really shitty high school microscope.
Where did they go?
You'll find it eventually.
Remember the story about that girl who couldn't find the thing?
Yeah, but there is a huge difference between 2 hours and 2 minutes.
Yeah, that noise is, yeah.
And he said, what, it pops once it reaches.
No, you just hear what sounds like explosions. Like, I don't think that's the sound it's supposed to make.
Area.
Have you found some of both?
Yeah, ish. We're not gonna do it until I zoom in all the way. But some of those little things. Yeah, that really annoying noise.
Ow.
Yeah. Okay, Okay, so the way that you do this, to go all the way down to the most magnification, at this point, just shoving a lens on there doesn't actually work. You need a little bit of like space filler to kind of guide the light through the thing. Like the air gap will actually screw it up. So you got to use what's called immersion oil, which is basically an oil that has the same refractive index as a piece of glass. So optically, it's basically independent.
But it's protecting you from the glasses.
No. It's literally just a completely optical path. So that the light will make it into the thing. The lens that we're going to be using is extremely tiny. So you just gotta have a little drop of this stuff. And it's actually pretty funny what this stuff is. It's distilled cedar oil. For whatever reason, that's just the ideal thing.
Refraction oil.
Yeah, you can use it. And traditionally with cedar oil, so it tends to be like a yellowy goo. And so old bottles of this stuff basically solidify as the cross-links. So,
This is basically like liquid fiber optics.
Like, so you just do on that.
So yeah, this will give you an idea of just how fun things are. They are really, really fun. Now, admittedly, I am damn near blind, so if you can't see things, let me know. But there you go. You'll see some are pink, some are purple. It's a pretty faint difference, but at the same time, you should be able to see it. Is it in focus? See? Yeah. Okay, good. Yeah, they're real tiny. Yeah.
Well, some are just like, no.
Yeah, some the bigger ones are more like globs of stuff. Yeah. When you're just fiddling with the fine control, it's able to like pick out the really tiny ones. They're really tiny. There's actually some bacteria that are so small you can't see them with this kind of microscope.
Really?
You need an electron microscope or some other.
Yeah. Called Mycobacterium. They're the bane of animal cell culture. Because they will contaminate your shit and they will screw everything up, and you're like, why is all of my tests coming out weird? And then you check and, oh no, it's Mycobacterium. All of the tests I've been running for the last 6 months are no longer valid. Yeah, that happened to my buddy who was working at the Mayo Clinic. Yeah, somehow Mycobacterium got into one of the incubators and just wrecked everything.
There's like a
There's 3 different Mayo clinics, and he was working in a lab in the Mayo, in the Florida. Because they're immune to most. You need a specific type of antibiotic to kill
Is there a
Mycobacterium.
stain to get them to show, or is there something?
There's chemical tests you can do for it. They'll have like certain. There we go. 10 minutes. So 10 minutes. So it is now 5:30-ish. So we'll give it 5 minutes, and that should be good. It'll pop a couple more times. Yeah, so that's that. Yeah, Mycobacterium are a goddamn nightmare. Sometimes if you get pneumonia, and like pneumonia that just won't go away, Mycobacterium.
So antibiotics won't do anything for it.
They will. You gotta have a very particular type.
But it's not like protocol to maybe just introduce that straight off the bat.
Yeah, because if you don't need it, it's better to have less antibiotics in your media because it can mess with your stuff. Because I mean, here's a fun one because I recently had pneumonia. And as I'm sitting there like gently dying, I'm like, wait a second, if mitochondria are bacteria, wouldn't antibiotics screw with your Oh yeah, they do. So yeah, I know it's a horrible noise. Yeah, so certain antibiotics can actually stress your mitochondria out. And if at any point mitochondria explodes, the entire cell kills itself.
Freak out.
Because it basically assumes that something absolutely horrible has gone wrong, and so it triggers cell death just to make sure that something like isn't going to make it worse. So my friend found out the hard way that one of the compounds in the pathway that makes things, in a particular pathway that makes things bioluminescent, very closely resembles a compound that is only found inside mitochondria. So the cells assumed that the mitochondria were exploding, and so seppuku, just in case. So that pathway was a no-go. Yeah, so that's it's just the kinds of things you stumble upon when you get doing this kind of stuff.
What was this compound that you?
I honestly can't remember off the top of my head. It was some weird Lux pathway. He was trying to get into zebrafish. He was wondering why the zebrafish kept turning into goo. Because unsurprisingly, they really didn't like that compound at all. Yeah, I think that's basically it. I hope y'all enjoyed.
Of course.
Yeah.
I'm gonna
Sign
be
me up
doing,
right away for the DNA modification.
I'm gonna be doing a fungi workshop next week. So it's gonna be shorter. So we're gonna be doing basically just like how you inoculate and prepare a jar of like fungi food, how you inoculate it and get the fungi growing, and then you get to take it home. And I believe third. I don't know. It's on the calendar. But yeah, so that will be baking fungi. And then you get to take home your jar, and then once it's ready, you basically just open it up and fungi will grow at the top.
You're inoculating questions.
Yeah, I'm gonna go to Micro Boutique and get just a couple different spore syringes so that they can use whatever they want. So probably get oysters, probably get probably lion's mane, because those are my favorite.
And they're eating.
Yeah, super.
Shiitake finicky to get to actually
I hate
produce.
shiitake. I love shiitakes, but I hate shiitakes. Growing is just such a pain. Oh yeah. Yeah. But yeah, lion's mane are delicious and they taste like lobster. So hard to go wrong. Real good. Not as good as matsutake, but matsutake are $12 a mushroom. So yeah, they're rare and they only grow in like one forest. So of course the Japanese are all over that shit.
You're gonna have to try and grow that here, I guess.
Yeah, good luck with that. Yeah, they're worth almost as much as truffles. Like they're it's the same idea. They only grow on their one particular tree.
Because it fits bacterial.
Yeah, just like the way they were, because fungi are also extremely symbiotic a lot of the time. Yeah, unless they're a rot fungus, like oysters. Oysters break down dying wood, but other things live in a symbiotic relationship with trees. So like, if you look at the roots of a plant, most of it is not actually plant. Like, it's mostly the fungus. So the fungus grows into the root cells and like makes itself homey. And so it turns off with the plant. So, no, it's still very flat. So the fungi give the tree phosphorus, and then the tree gives them sugars. And so they like live off of each other, and then the fungi will connect trees, and it can like pass stuff between trees to communicate.
Virtually impossible to recreate that.
Yeah, you gotta have to.
Perform that.
Well, so I mean, you can farm truffles, which is a misconception because they say you can't. You totally can. You just need to uproot the entire fucking tree.
Okay. So you can farm truffles by basically moving it out of its natural environment.
And then putting
And
it into a
taking the
grove,
environment.
and then like spreading some of that soil, like getting all the same tree growing, and then it will actually spread.
You can't do that.
Yeah, with sterile soil and everything.
Yeah, well, I mean, and it takes 20 years, so you know.
So you have to figure out which exudate the tree produces that is feeding the mushroom.
It's a nightmare. Fucking nightmare. You can do it. People do farm truffles that way, but
Soils
farm in the
are
sense of you spend
California.
20 years making your farm, and then at some point you go around the truffle and dogs and see if you got anything.
I don't think that's allowed to be farmed at that point.
Yeah, I mean, technically, it's like farming cork. You can only harvest it once every 20 years. But who's got that kind of time?
Well, no, we're not out of that, of course.
Yeah, exactly. Yeah, it's interesting. They've switched from truffle hunting pigs to truffle hunting dogs because the pigs want to eat the truffles. The dogs just will just find them. So it was a lot harder because once the fucking pig found one, you got to hold the thing back while you get the truffle and then hide it from the pig. And it's like, hey, I did all the work. I want my truffle.
Imagine how many truffle hunters are just like, good meat.
Yeah, and you're bacon.
Yeah, that's it, buddy.
You don't find any truffles and you're dead. But no, now it's dogs because you train the dog to just sit, not eat the truffle because they don't care about the truffles, but you just trained it to find them.
So it's like over time, I might find less, but like only eat someone.
Yeah, something like that. That's fine.
Apparently it mimics the female hormone, so the pigs get horny.
Yeah. God. Yeah.
There's no reasoning coming.
How we doing? All right, so that's been about 5 minutes. The nice thing is, once this is at pressure, it'll stay at pressure because it's so hot. So just letting it cool slowly is actually sufficient to get it to fully be be sterilized.
Is it one of the good ones that forms a vacuum when it cools down? Does it stay like sealed when it cools down?
I mean, it will stay sealed, like until I open it. Basically, it's not going to form a vacuum, but it will stay clean. So until I open it, it is sterile. And because there's no gel in there, I don't care about opening it tonight. So I'll get to it when I come back to the lab. Yeah.
Awesome. Well, thank you very much. That was
Very welcome. I hope you guys enjoyed. We will do the genetic engineering one hopefully next month, if not the month after. We just got to plan that out.
So you're really rolling out all the workshops now?
Yeah. And then we're going to do this on a circuit. So we'll do this one again.
Oh, yeah?
Yeah.
Okay.
So if you want to see me do the same shtick over again.
Ask the same questions.
Yeah. What is a protein? Wait, I'll get that. But yeah, we're going to do them on a circuit. We've got a couple genetic engineering ones coming. We're thinking of doing that workshop as basically like an entire, like a long workshop, like 5 hours. You do the entirety of the procedure. So you isolate the gene that you want from one piece of DNA. You do DNA extraction. You put the piece of DNA that you want into the other piece of DNA. And then at the end, put that thing that you build into some yeast and make it glow red in the thing here.
That sounds pretty good.
Yeah, then everyone would go home with their own like handmade
Crazy
genetically
deer.
modified.
Really?
Yeah. Because why not? Yeah, that's
Few
it.
reasons why not.
I mean, yeah, so that's basically it. If you guys want to hang out, you're welcome to do so. There should still be some glowing meat on tap. If that's the
Love
thing
to
that you
try
would
that.
like. There are mason jars kind of floating around. Just grab one. There's a bathroom down the hall. Just give it a quick clean. Because fuck knows what people left in them. Because people are the worst.
What did you say the washroom?
Just down the hall.
Down the hall on the left there.
But yeah, so I'm gonna just be tidying up. If you got any other questions, you're welcome to ask. I'm gonna check on the live stream and see what sort of cesspool the comments are. Because oh my god. Love what? Yeah. Okay. What's happening? Okay. All right. Well, thanks for coming. Bye.